floor to a very important person for all three projects. She guided us in a very nice and friendly way through our project's time. And let's welcome on stage Chloe Johnson from the Bio-Based Industries Consortium.
Thank you very much for the introduction, Kevin. So I also have a few notes to make sure I don't forget anything. So first of all, it's a real pleasure to be here at this final event for Bizente, for Recover, and for Enzycle. So three brother projects that have a very important part in our portfolio of projects. So first of all, good morning to everyone and welcome to this event. As project officer of these three projects, it's really an honour for me to open this event and to address all of you. And it's a really great opportunity not only to meet with all of you who have been working together with me for the past few years, but only managed to see many of you on the screen. So it's a real pleasure for me to meet you now all in person. All of you have been committed for the last 4 years of these projects. But also, it's a really nice opportunity as well to be part of this very interesting program that the organisers put together. So as Catherine explained, there's not only a chance to showcase the results of all of these research and innovation projects, but also how this then interacts with industry, with policy, and also ample time for discussion about some key themes, which no doubt will generate some very important conclusions. So I would like to say a few words now about these three projects that have brought us all here together today, about Enzycle, Recover, and Bizente. But if you'll allow me first to quickly say something about Circular Bio-Based Europe joint undertaking, because in that way I can explain why these three projects have such an important place in our portfolio. So Circular Bio-Based Europe joint undertaking is where I'm project officer. It's a 2 billion public-private initiative between the European Union, represented by the European Commission, and the Bio-Based Industries Consortium, which are the private partner representing the bio-based industries. So the main objective of CBEJU is to help build a competitive, sustainable bio-based industries in Europe. And how we do this is to help to de-risk investment. And so concretely by funding innovative projects such as these three projects we're here to celebrate today, and helping to really advance solutions such as new bio-based materials, products and building blocks, but also, as we'll see today, also biotechnological tools that will help us solve some challenges of today and the future. But it's not just to advance these developments, but also to bring them out of the lab and closer towards market. So you know that we have a very special mechanism to fund also flagships, which are at a very high TRL, close to, in fact, commercialization. But what kind of innovations are we talking about? They could be, for example, new bio-based textiles, new packaging solutions, construction materials, to name a few. But overall, the main ambition is that through our projects, we want to provide circular solutions to the problems of the economy and to move towards more sustainable models of production and consumption. And that's where really there's a strength to these three projects, Recover, Enzycle, and Bizente, because they responded to a call in 2019, the same topic, to resolve the end-of-life challenges of non-biodegradable and recyclable plastics and composites. And they're all bringing their own solutions and their own to their own unique challenges. And we all know here today that this is a very important challenge because at present, the latest statistics I read is that only 1/3 of plastic is recycled in the European Union. So clearly we need new disruptive solutions to deal with this challenge. And that's what these projects are offering. So on one hand, we have Bizente, that's tackling the challenge of thermoset composites, how to degrade these materials that are currently mostly going to landfill, not recycled. But also how to valorize the resulting new building blocks into new products, for example. Then we have Enzycle, that's tackling the recycling of difficult to recycle plastics. For example, post-consumer plastic packaging waste, but also multi-layered plastics, which, as we all know, is a challenge because you have the issue of separation. So Enzycle has been really bring a new opportunity there. And then last but not least, we have Recover. That's responded to the challenge of dealing with difficult to recycle, for example, agricultural waste products, but also packaging, and using a novel combination of enzymes, earthworms, and insects and microorganisms to do so. So I'm proud that these 3 projects are counted in our portfolio of 163 projects to date. Firstly, because they're helping to build a circular economy by valorizing traditional waste streams, and doing so not only generating new economic opportunities, but also ensuring that this non-biodegradable waste does not end up simply discharged in the environment or landfill. Secondly, because you're all advancing solutions that are helping us to deal with environmental challenges, for example, microplastic pollution. So helping to protect our soil and water by giving new solutions and offering new solutions to incorporate enzymes or microorganisms into processes like wastewater treatment, but also composting. But thirdly, because you're also contributing significantly to knowledge creation and scientific breakthrough, particularly in the field of biotechnology. So to date, already 21 publications in this area. But finally, what's been a real recipe for success in the case of these 3 projects is how you've put together and how you've really embraced the complementarities and synergies between the projects. I think this has been a huge strength and something that I've enjoyed watching as you put together through webinars and through different joint events, but also offering a lot of knowledge sharing. I'm sure that this will trigger a lot of fruitful collaborations also in the future and new and interesting projects. So I want to end this by saying congratulations to all 3 projects, to Enzycle, to Recover, and to Bizente, and to thank you for your great commitment over the last 4 years. So to all partners, but in particular to the coordinating partners, to Atenic, to ITIM, and to the University of Almeria, and more personally to Marta Redrado, to Juan Antonio Tamayo, and to Maria Lopez for your excellent guidance and how you really kept the ship sailing, but also maintained an excellent communication and collaboration with me as well. So all 3 projects have brought important advancements, insights and lessons learned, and I really believe this is bringing us closer to a circular future. So thank you very much.
That was summarized so nicely that I can skip some parts of my text here, and I give the floor to Bizente. Marta Redrado Notifuri from ITIP. Sorry. Okay, so all on stage, all coordinators on stage. No? No. We start with you, so you have the first presentation.
Are we recording? Are we recording the event? I'll check. I'll check. Sorry, 1 second. Okay. Yeah, it's recording, so it's all good. Yeah, this one is mine, and then I duplicate here. Yeah, it's perfect. Thank you. Hello, everyone. No, sorry. Now I need these microphones. Now, can you hear me? Well, yeah, good. Good morning, everyone. Thank you, Chloe, for your nice words. It's been a pleasure, as Catherine said, to work with you over the last 4 years. And thank you, everyone, for joining today to this final event, this celebration that we are concluding our projects. As they say, my name is Marta Redrado. I'm the coordinator of Bizente, of one of the 3 projects. And I work at ITIP. We've been coordinating for the last 4 years, Bizente. What I'll try to do today is to very briefly explain you what we've achieved in the last 4 years. But so you understand where we're coming from, I'll present also a little bit why Bizente, the overall of the project, as well as some of the technical details of what we've achieved, and of course, what impact we expect to have with our project. So why Bizente? Why we thought this could be a good idea? It's because we saw that thermoset plastics are becoming a challenge for the waste management industry, in particular those involving the thermoset composite materials. These are currently they are not recycled, they are either incinerated or diverted to landfill. So that's mainly due to their inherent complexity and the lack of or the difficulty in applying eco-design techniques. Hello? Someone online, maybe? Okay, so because we found this problem, what we try to do is what we propose, it's a solution that to find for these end-of-life issues of the thermoset composites, that it was based in a new technology based on enzymatic and control enzymatic biodegradation. The objectives that we put to ourselves in the project is to demonstrate this potential of the enzymes to solve the problem of the end of life, then develop this technology in a pilot environment, and finally establish a new value chain for the recovered products after the biodegradation. For that, we counted with 10 partners from 5 different European countries with complementary expertise. We counted with 3 research and technology organizations, 5 small and medium enterprises, and 2 large companies. Before I go into the details of what of each of the technical steps that we achieved, I want to give you a very general overview of what Bizente was or is still. And it's that we started from commercial materials, and what we did in parallel is, on one hand, the commercial enzymes, we customized them to improve them and to improve the biodegradation process. And then on the other hand, we did the same with the resins. We did some modifications, so they will also favor this biodegradation process. Once we have them both at lab scale, we use model compounds to simulate this and also to find out what the products of this reaction would be. Once we had that first stages, what we did is to move into the bioreactor, and for that we used thermoset composites that the industrial partners gave us from both the construction and the aeronautical sectors, and we pre-treated them in 2 ways, chemical and mechanical, and then we put them in the bioreactor and studied the whole process. From there, what we got is some products that they were recovered, and on the other hand, we wanted some monomers that they could be reused, and we studied what other life we could give to these monomers to introduce them back in the market. Of course, at the same time, we had the transversal activities that went along through the whole project, like, for example, the dissemination, communication, and exploitation activities, and we also did some toxicity, LCA, and LCC studies. If we move on more into the detail, first of all, I would like to say I'm not the technical expert in any of what I'm going to explain now, but we have the whole consortium here who could raise a hand if I'm saying something that is a bit crazy, or maybe answer your questions later that I might not be able to answer for you. So in the field of the resins, what we did is we studied 4 different resins in the project, 2 epoxy at the bottom, and an unsaturated polyester and a vinyl ester. The idea is to introduce reversible bonds to these structures, and for that, the strategies that we follow is for the unsaturated polyester and the vinyl ester, very similar, and is to replace the styrene by the vinyl ester in the case of the polyester resin, and by methacrylates and disulfides for the vinyl ester resin. For the epoxy resins, what we did is to remove the amine hardener by disulfide compounds. The 4 resins were studied. For the 4 resins, different processes and iterations were carried out to modify them. And what we based these modifications in are in the thermal properties, which are quite important, especially the TG, the glass transition temperature, that they had to be as similar to the reference resin as possible. With that, you can see here one of the, for each type of resin, you can see a resin that we developed in the project. And we also studied rheology and as well as the chemical degradation. As you can see here, for the R930 and the Aragen, the chemical degradation was total for both resins. It was 50% for the RTM6, and for the dione it was 5%. And also it has to be noted that for the R930, some enzymatic biodegradation was also seen. We move into the directed evolution of the enzymes. We tested all types of enzymes to work in all types of the resins we had, but in the last efforts were put into the ester resins. They were the ones targeted because they seem to have better chances to work. So these are the two there, the dione impact and the R930, the ones that you can see that we put our latest efforts in. To start the directed evolution platform, we had to select a model molecule, and that was the p-nitrophenyl 3-methyl acetate, which it was selected because it's got a structure pretty similar to the one of the ester resins. It was used for the HTS, the high throughput screening, and it was what it gave it was really easy to follow the hydrolysis because it releases a yellow, well, a colorimetric compound that they can be followed. So once this screening method was developed and was validated, then the directed evolution campaign started using the catinase Fusarium solani as the parental enzyme. And after four generations of mutants and combining several protein engineering techniques, it was identify the protein P6, which it's got a great thermal stability, and also it's got a really good activity, being 120 times better in biodegradation than the parental enzyme. Directed evolution campaign followed two approaches. The first one on the left, based in improving towards the model molecule hydrolysis. And the one on the right, it was more concentrated the efforts in finding the thermal stability, which is one of the key points for the enzymatic biodegradation. And finally, or after a lot of efforts, they all converge in this P6 that I just mentioned before. That's the final mutant after 4 different mutations, and as I already mentioned, with a great activity and very good thermal stability. Once we had this, well, and in the meantime, not only for this one, but for the different enzymes that they've been developed, they had to be produced in an industrial relevant environment. For that, what we did is a lab scale. We first studied a lab scale and then moved into the industrial scale. We studied the expression of the yeast cells to define a scalable production protocol. In a 6.5 liter reactor, what we identify is how the protein expression responds on the basis of the applied cultivation strategy. Once this protocol was found, the best production was defined. The scale-up activity consisted in developing fermentation and downstream procedures from this 6.5 liter reactor to 150 liter bioreactor. This step, then what it did is push the production of enzymes from 10 in only one batch from 10 grams to 230 grams. At the bottom right, you can see the workflow diagram of the whole process. Also, I would like to add that in the last few months, what we've tried to do successfully, by the way, is to remove the methanol from the process. So then it's more environmentally for both the environment and also safer for the people working on it, but it's also more economically viable because we remove one of the components. In parallel to this production at industrial scale of the enzymes, what we did is study the enzymatic biodegradation. For this study, the 4 different types of resins were covered, and as I already mentioned, the best results were given for the R930, for the polyester resin. What we based, or what not based, but what we saw is that in this process, one of the main products from the enzymatic biodegradation is the isophthalic acid. You can see at the top right a chromatogram showing the increase of the isophthalic acid signal. In order to understand better the reaction mechanisms and improve the degradation yield, several approaches were followed, and those ones study, for example, the ratio, the optimal ratio between the resin and the enzymes. Also, we study how the pretreatments, both chemical and mechanical, affected the process, as well as what's the influence in some variants of the enzymes in the process. For example, the temperature. Thanks to this P6, we could see that reaching higher temperatures will improve the process, and we could achieve it thanks to the last mutant. But also how long the process needs to be working in order to reach the peak. After several of these testing these several approaches and a continuous improvement, a maximum release of isophthalic acid per gram of resin treated was defined, as you can see in the graph at the bottom right. This is a very visual graph of how different treatments, how different enzymes, how different processes will change the amount of released isophthalic acid, getting best results with, of course, the optimized process, the micronized resin, and with the high temperature. What we did as well, it's one of the last few steps, and I have to say we are going to present very general results, as some of them are still ongoing, and we won't have our results. Well, we will have them maybe now or maybe next week, but I will present some preliminary results for the toxicity evaluation. For this test, what we picked is organisms that you can see them there on your left for both land and water environments. And we check some endpoints, some toxicity indicators. We check the mortality, we check the behavior, and also we check some biomarkers that they give us an idea of that there's something wrong with these microorganisms before we can actually see it happening in them. These toxicity tests were tested in realistic environmental conditions and well in some relevant scenarios. Here you can see a small summary of what we've achieved so far. As I mentioned, R930 is the resin that gave best results in the enzymatic biodegradation. So the toxicity tests have been carried out for the whole family of these resins. So starting from the commercial resin to the released products like the isophthalic acid. For the commercial resin, both organisms, the fish and the tenebrio molitor, they both didn't show any sign, any negativity, so it's all good. But then when we move to the modified resin, although the worms and the tenebrio molitor is fine, we could see maybe some stress in the fish that I actually don't remember the technical names of the fish. But here we have the experts in case you want to know. When we tested the chemical degraded resin, we could see also good results for the worm and the tenebrio molitor, also some stress in the fish, and it has to be noted, although it's still not conclusive, but the preliminary results show that in the artemias, when the concentration of this resin, chemically the modified resin, is above 0.85 grams per liter, there was a mortality in 50% of the population, okay? And the last step, that is the last one, because it's after the biodegradation process, is the one that has less results up to date because it's what is being tested at the moment, is the enzymatic degradation products. And this includes the enzyme, liquid and solid fraction, the release products. So to give you like a brief overview, we are just going to concentrate the results in the isophthalic acid, which is the acid that is being released, but for the commercial that we bought, to simulate the same results. For this, we saw that the tenebrio molitor is fine, but for the worms and the artemias, we could see some, some deaths, yeah, when the concentration of isophthalic acid is about 0.5 grams per liter. But the positive results is that in our process, in our biodegradation process, the concentration of isophthalic acid is less than that, so then there shouldn't be any deaths, and that will be confirmed in the next few weeks. So this is, I just gave you a very, very quick overview of what we've done in the last 4 years, of all the partners, what they've tried to achieve. And with that, with all this effort that was put into it, what we expect is that Bizente will contribute to decrease the amount of thermoset composites that are released to the environment in at least a 40%. We also have some preliminary results that the CO2 emissions are going to lower from 20 to 35% due to using less fossil-based raw materials. And also the technology being developed is expected to have a low demand. And with that, I will conclude my presentations, but I wanted to thank in front of everyone the consortium of Bizente, because they've worked amazingly and beyond expectations to achieve what we've achieved so far. So I wanted to thank them all because it's been a pleasure to work with them. And I don't know if, Catherine, the questions are now or later.
I think we have time. We can take 1 to 2 urgent questions and then have a discussion round at the end with all coordinators on stage as well.
Perfect.
So if there are any questions for now, yes?
Martha, David Newman, thanks so much for that presentation. Can you go back 1 slide? You say that this will allow the amount of plastics to be diverted from landfill incineration by at least 40%. But you're assuming, therefore, that those plastics will go through this process. You're assuming, therefore, that this process will be adopted on industrial scale.
Yes, that's what we hope.
But how many years are you away from that?
That we are still far away. We did some business development, and this is a project that is in a very low TRL, like basic TRL, from 3 to 5. So we did some calculations, and we expect this to be in the market in the next maybe 10 years.
Another question? I cannot see any hands. Then I think
I can do that. Tommaso De Santis, did you do any economic evaluation on the process, or I don't know if that is?
Yeah, no, sorry. Thank you for the question. It's out of the. We didn't present it today because the partner who is doing it told me that he still doesn't have conclusive results, so he wouldn't want to present anything that is not like really true. That's why. But it's part of the project, and in the next few weeks we will present it in the report. Of course, I can give you like what we've seen so far is that actually economically it's more like the new resins are more expensive. But then, like always, you know, everything that is new and that it's usually bio or more environmentally friendly, at these days it's a little bit more expensive or more expensive than what is available in the market now. But we hope that the positive things go against this maybe difference in price.
Okay, thank
you. Then I think we move on with the next speaker, which is Juan Antonio Tamayo Ramos from Italy, who is representing the Enzycle project.
Good morning, everyone. It is a pleasure to have you here. Thank you for coming to this final event of our 3 projects. My name is Juan Antonio Tamayo. I am the project coordinator of Enzycle from ITENE. As Chloe mentioned in her nice introduction, Enzycle, together with Bizente and Recover, was funding in the 2019 topic Applied Microorganisms on Enzymes to Solve End-of-Life Issues of Plastics, aiming to convert and remove plastics from the environment. Some facts about Enzycle. Well, Enzycle is coordinated by ITENE, which is a research institute located in Valencia, Spain. It started in June 2020. It will finish next month. It has an overall budget of 6.4 million euros with a substantial in-kind contribution from different companies that you see on the slide. This is the Enzycle consortium as a whole. We have a well-balanced consortium of universities, RTOs, and companies, both SMEs and large enterprises from all over Europe. And well, I will start my presentation talking about something that everyone knows, right? I mean, most of the plastics that are produced nowadays in Europe, but also in the world, are fossil-based. Only a small part of these plastics come from recycled sources, and even a smaller fraction or portion of these plastics are bio-based. So, well, we still have a long way to go towards more circular production of plastics. And well, then we have to tackle the waste management issue. As Chloe mentioned, currently we are only recycling more or less 1/3 of the plastic that we consume, while other options less sustainable, such as energy recovery and landfill, are not desirable, right? An interesting fact is that when we separate plastic, indeed, we enhance its recycling. So when we separate plastic, we enhance recycling by 13 times, which is very good. But even when we separate plastic, we cannot recycle all the plastic that is separated for different reasons, right? So this is the problematic that Enzycle was aiming to tackle. So we focused in particular in some plastics, mostly coming from packaging, that cannot be recycled currently, such as multi-layer packaging, post-consumer PET trays, and clamshell containers. And well, Enzycle was also aiming to provide new solutions to reduce the presence of microplastics in the environment, and in particular in wastewater. Enzycle focused on the development of technologies based on the natural ability of some microorganisms to degrade plastics. So it is, of course, a good example, the one of bacteria that are able to secrete enzymes, glutinases, also called polyester hydrolases, that are able to degrade some types of PET. So these polyester hydrolases, they hydrolyze PET into its constituent monomers, namely terephthalic acid and ethylene glycol. So based on the ability of these microbes, well, we set as a goal for Enzycle to identify new enzymes able to degrade not only PET, but also polyolefins, in particular polyethylene and polypropylene. So the idea or the goal was to identify new enzymes so we can develop new processes for the degradation of these plastics. So once we have identified promising enzymes, well, we are producing them up to pilot scale. So we can develop also degradation processes, also up to pilot scale. I'm talking about TRL 5, 6. So once we have our efficient depolymerization reactions working well, we could, on one hand, eliminate the plastics, and on the other hand, process the plastics hydrolysates to recover the constituent monomers. So we could reuse these monomers for the production of new products, such as new PET materials, polyurethane, and also, for instance, low-density polyethylene films. So how did we do that? Well, we started, as I mentioned, looking for new enzymes. So looking for new polyester degrading enzymes, and also trying to find new microorganisms that could have a role in the degradation of polyolefins. So talking about polyester degrading enzymes, we went to different environments to pick PET residues. We isolated metagenomes that were present there from bacterial communities, aiming to pinpoint or identify polyester hydrolases. So we applied some in silico analysis, some hidden Markov models, etc., to identify new genes coding for polyester hydrolases. And we selected some candidates so we could find enzymes that would be efficient in degrading PET. This was for the PET. For the polyolefins degradation, we followed similar but different strategies. So we aimed for the identification of microorganisms, also microbial consortia, that were naturally growing or present in, for instance, polyethylene and polypropylene. So what we did is we isolated these new strains from polyolefin residues. We isolated these bacterial strains consortia, and we enriched microbial cultures in minimal media containing polyolefins as sole carbon sources, so to promote the development of these particular microbes or microbial consortia able to use these plastics as carbon source. Once we would have our microbial isolates, what we did is we studied their genomes or metagenomes in case of microbial consortia to identify as well some enzymes that would be produced by these microbes and would have a role in polyolefin degradation. So we were successful in the identification of both enzymes with potential to degrade PET and polyolefins. Regarding polyolefins, we were able to identify some enzymes with some capacity to, or microbes with some capacity to alter the structure of polyolefins, but we were not really efficient in degrading these plastics. So we, in the end, put more effort in scaling up processes for the degradation of PET, both in monolayer PET and PET polyethylene multilayer materials. So the rest of my presentation will focus on the development of the technologies in scale-up processes for the degradation of PET and PET polyethylene materials. So as I mentioned, we identified PET degrading enzymes from different families. In the end, our goal was to select the most efficient enzyme for the degradation of PET, so we could use this enzyme in scale-up processes. And from all the enzymes that we identified, well, the most promising ones were produced both in E. coli and Pichia pastoris, so two expression systems widely used for the expression of recombinant enzymes. And finally, we decided to focus on one enzyme, or actually we focused on two, on LCC and PHL7 enzymes, both of them able to degrade PET. But we did most of the job, so for instance, protein engineering with PHL7, because in the end it was the enzyme that was more promising in terms of activity. It is a highly active enzyme against PET, and well, we studied the most relevant residues providing this enzyme this particular high activity, thermal stability, etc. Once we identified the most relevant residues, we would be able to modify them, okay, to improve the characteristics of this enzyme, which we did. And for instance, we were able to obtain new variants of this PHL7 enzyme, which showed better activity rates and also a higher thermal stability, which are features highly desirable when we are using these enzymes in PET degradation processes. And with these efficient enzymes, we managed to have reactions as such, so where we are able to degrade, to fully degrade PET trace in a relatively short time. So this is very good results. So once we have these results at laboratory scale, well, it was time to scale up the operations at pilot scale. So we started producing the enzymes at pilot scale in fermenters up to 300 liters. We produced the enzymes both in E. coli and Pichia, also at this level. Once the enzymes are produced at pilot scale, we can run also reactors at pilot scale. We have been using a reactor of 120 liters, where we can put significant amounts of plastic. So we can also produce significant amounts of plastic hydrolysates, which we need to continue with our downstream processing operations to produce enough monomers, so we can also try to produce new materials, right? Which was the initial goal. So with our depolymerization reactions, we produced indeed hundreds of liters of PET hydrolysates. In the beginning, we were aiming to develop downstream processing for both TPA and ethylene glycol, but this was proven to be too challenging. So in the end, we focused on the purification and crystallization of TPA. We tested different protocols at lab scale, but for a pilot scale, we employed supercritical CO2 technologies for the purification and crystallization of this compound. And we have successfully produced kilos of this material, which are transferred to companies that are testing their suitability for the development of new PET materials and polyurethane foam. In terms of PET repolymerization with recovered TPA, this has been, quite a challenge endeavor because, you need to have high quality and purity TPA to obtain a good quality PET. So one quality characteristic of PET, for instance, is color. So as you can see in the screen, we have a gradient from trial 1 to trial 3 in terms of color. The first trial, a bit brownish, and then the more we iterate to improve these purification crystallization protocols, we get a PET which has a whiter color, which is a quality indicator, right? And in terms of polyurethane production with TPA, the goal has been to substitute phthalate acid, which is used to produce polyols that in the end are used for the production of polyurethane. So the goal has been to partially substitute this phthalate acid with terephthalic acid. Okay, so we have been testing different substitution rates. So as you see in the slide, 50-50 substitution in red and 70% substitution in blue. What we have seen is that when we substitute 50% of phthalic acid for terephthalic acid, we obtain a polyurethane which has the same characteristics as the 100% phthalic acid. So we are using this recipe to produce polyurethane containing recovered TPA from the depolymerization process. And as I mentioned, we have also focused on the depolymerization of PET polyethylene trays. So we have not been able to tackle the issue of enzymatic depolymerization of polyolefins, but what we can do is we can eliminate the PET fraction from a PET polyethylene tray, which is good because if we eliminate the PET, we end up with the polyethylene. And once we have the polyethylene, we can do mechanical recycling with it to produce, for instance, films. So this is an example of an starting material on your left. We started with, for instance, 4.5 kilos of PET polyethylene material, and with our enzymatic reaction in our reactor, we were able to eliminate virtually all the PET present. So we would recover 250 grams of residue, which contains mostly PET polyethylene. And I say mostly because if we virtually eliminate all the PET, but you always have some residue of PET there, which is a bit annoying, but well, it is what it is. Probably some of the PET is not really accessible by the enzyme, or maybe the presence of the adhesive is also inhibiting the accessibility of the enzyme. We don't know yet, but so we obtain this residue. When you try to convert this residue in a polyethylene film, okay, you get a film, but this is a low quality film because there are some PET residues which are interfering. So the melting point of PET is 265 degrees. The melting point of polyethylene is 195 degrees. So well, it is not the same melting temperature, so it's complicated. So what we decided was to perform an alkaline hydrolysis of the polyethylene residue to eliminate this small amount of PET that was still present there. So we managed to obtain a clean polyethylene material, which in the end it was indeed suitable to produce nice thin polyethylene films, which is a very nice result. As you see, the film is transparent, so it has also quite good physical, mechanical properties. So we are pretty satisfied. And last but not least, as I mentioned at the beginning of the presentation, we were also trying to tackle the issue of the presence of microplastics in wastewater. We have a company, DAM, putting or contributing within Enzycle with their wastewater treatment plant. They developed a system to recover the microplastics from their wastewater plant. Composed by a hydrocyclone to concentrate the microplastics, aeration system to separate the microplastics by flotation, and also a filtration device, which you see surrounded in blue, with different meshes of different pore sizes, so we could separate the microplastics by different sizes. So this allowed them to recover the microplastic fractions present in the wastewater in their facility. And we also performed, of course, some analysis to identify which compositions of the microplastics were present. So we found out that most of the microplastic present was polyethylene, and there were also fractions of polyamide, PET, and PVC. So what we did with these microplastics, once they were recovered, was to subject them to different degradation systems. Actually, we were not able to recover so many microplastics from these wastewater treatments, so we were using a microplastic mix, a modern microplastic mix that we were producing in Enzycle. So one of the systems that we employed for the degradation of these microplastics was to subject them to a pretreatment, and then to subject them to an enzymatic treatment with our polyester hydrolysis, with a subsequent biological treatment employing microorganisms that had shown some ability to alter the structure to partially degrade polyolefins. We developed this system to be implemented in DAM. And we indeed observed the degradation of microplastics at some degree, but well, to be honest, we were successful in the degradation of basically the PET fraction of the microplastics, right? We observed some modification of the structure of the polyolefins present. We analyzed these results with Raman, and this is how far we could reach. It is problematic when we are dealing with plastics that are not PET, and when you have plastics with high crystallinity. So, well, we are satisfied as we have done some progress, but of course we need to keep working on it. And we also tested the degradation of this microplastic consortium model in industrial composting and anaerobic digestion processes. We did these processes also employing our polyester hydrolysis to see if there could be some synergistic work between the microbes naturally present in these environments plus the enzymes, and indeed we were able to see some degradation of the microplastics, same as what we did in the microplastics degradation system. But well, we also observed in general low biodegradation efficiency. So same as said before, we need to keep working on this to improve the degradation rates. Besides the technical work or the laboratory work, we did as well some safety, environmental and techno-economic assessments to evaluate the sustainability and the economic feasibility of the new processes being developed. And for instance, within the techno-economic assessments, we were able to identify which are the necessary conditions that need to be accomplished in order to have an enzymatic process as techno-economically feasible as what it is currently, for instance, the mechanical recycling for PET. We have learned a lot in this sense, and we will also report it very soon. And regarding our dissemination, exploitation, and communication activities, well, what we aimed was to share our activities to different stakeholders, for instance, to promote awareness and also to tackle the societal issues that are related to the plastic recycling problem, aiming as well to offer the technologies developed for their exploitation. So we have defined exploitation roadmaps, and actually some of the Enzycle results have been already transferred to other European Union and Commission projects that are recently started. So the results that we have produced will be the base for further research, which will allow to continue progressing. From my side, I would like to end my presentation also thanking the Enzycle Consortium. We have been a very fruitful consortium. We have successfully achieved most of the goals that were established at the beginning of the project. Thank you all for your nice collaboration. Also, thank you all for your attention. And yeah, any questions, please go ahead.
Thank you. Any questions? No hands? Then I think we can move on with the Recover project, Maria José López from the University of Almería.
Hello, everybody. Can you hear me? Well, thank you all for being here in this intense journey, because we have 3 projects with a lot of results, and we are at the end of our 4-year way. So I'm Maria José López. I'm the coordinator of Recover, and I'm a professor from the University of Almería. Well, in Recover, we develop innovative biotic symbiosis for plastic biodegradation and synthesis to solve the end-of-life challenges in the agriculture and food industry. Just with the title, more or less we can see what is this about. So in my talk, I will first introduce the project, the global problem, which are the plastic and the objectives, and I will show you the results that we obtained from our actions, and finally conclusion about this project. Well, Recover started on June 2020 and will finish on May this year, and is composed by 17 partners from 7 countries across Europe, and the total financial support is 5.8 million euros. What is here, this is about 3 paths, 1 solution. And the paths that we have been exploring in Recover involves the combined action of something bigger than the enzymes and the microorganisms. Together with microorganisms and enzymes, we have been working or combining the activity of microorganisms and enzymes with insects and also with earthworms in different scenarios, as I will show you now. And we focus on the plastic that are used in the food packaging and for food packaging and in agriculture. These two sectors, agriculture and food packaging, consumes about 40% of the total production of plastic, which accounts for about 350 million tons. But as has been told here today, only one small fraction of this, around 31% of the plastic, is recycled, and most of it is incinerated or disposed of in landfills. And a big part of this plastic, more than 50% of food packaging that are collected in the municipal system and the waste management system end up on landfill or also in the compost, and then goes to the soil, so it contaminates the soil. And in addition, also mulching, some practice in agriculture like mulching, but not only mulching, but well, especially that one is also suspected to be a source of plastic and microplastic in terrestrial environment. So just to know what we call agri-food waste plastic, and which are the recovery scenarios, which are the plastic that we are focusing on during the development of the project. We focus on the non-recyclable fraction of the plastic that are collected separately but cannot be recycled because they are mixed, or they are a mixture of different types of plastic that are difficult to recycle. So this is what we call, well, this fraction usually is incinerated or landfilled. See, this is one of the scenarios where Recover is trying to, or has been trying to find a solution. This is the direct disposal of plastic, mainly made with food packaging. This is one of our fluxes, right? And then other part of the plastic goes with the organic fraction in the waste management system, where usually this organic fraction is composted, usually when it is collected separately. This is the other scenario for recovery, is the organic waste, which is polluted with plastic. And finally, part of the plastic that, well, it is directly released to the environment and goes to the soil, or the soil which is contaminated with the mulching, or because of the use of compost contaminated with plastic. This is the other scenario, the other real scenario in which Recover has focused the work, which is agricultural soils polluted with plastic. And so I will say later, we focus on polyolefins, which are difficult to deal with and to degrade by biological means. I think after this, this is something that is constant for all the project, because when we are dealing with polyethylene, it is not that easy to perform a biodegradation. So in Recover, this is the general concept of the project. We develop novel biotechnological solution for this non-recyclable mixed plastic, and we use a combination of microorganisms, insects, earthworms, and also enzymes to convert the agri-food waste plastic stream into biofertilizer and bio-based material, which are used also for food packaging and for agricultural applications, or closing the loop. Action that we have more or less or almost accomplished during this 4-year project can be schematic like this. First, we focus on the problem. We characterize. First, we have to select in which polymers we were going to focus on, and we characterize them and define the collection strategy and the pretreatment options, focusing on municipal solid waste and also agricultural plastic in general. Then we selected the tools that we then use it to transform or to degrade completely, if possible, the plastic. The tool we selected and biotechnological tools, those microorganisms, enzymes, earthworms, and insects, and we upscaled the production of those selected biotech solutions. And then we upscale it, we transfer this biotech solution to real or more or less close to real scale environments, these three scenarios that I started with. Also, this is the solution, upscale and monitor the biodegradation capacities that we tested for bioremediation in soil, for in situ bioremediation, and ex situ, where the plastic were treated well in compost and vermicompost treatment, and also by insect breeding in the plastic. Then we also have analyzed the value of the downstream products. We develop biofertilizer and chitin-based bioplastic and coatings that comes this chitin from the insects, and also have analyzed the sustainability and the impact of the project in different parts. So regarding the problem, first we had to select. There are so many polymers in the different waste fluxes that I have mentioned, but we finally selected, I think, a lot of them, because we selected four polymers for plastic, which is polyethylene, linear density, low density polyethylene, and linear low density polyethylene, LDPE and LDPE. You have the structure there, and polyethylene terephthalate and polyester on the basis of the use in the food packaging industry and also in agriculture, and the potential biodegradability, because there are other polymers that are even more difficult to biodegrade than those that we selected. And we consider not only the virgin, the virgin plastic, but also the recycled one. That was the plastic that we use, all the consortium use for the different experiments, either in film or in granules or micronized for the different experiments. And we also analyze in municipal solid waste and also plastic coming from the agriculture, more than 40 samples that were analyzed, and at the end we found that they were mainly composed by multi-material and multi-layer, which is another block for the recycling of the plastic. But they had quite a big amount of polyethylene inside those plastic. Also, we use different techniques to differentiate the plastic polymers, and in fact, one of the systems that was developed, has been developed by one of the partners of Recover, was a system that is capable to differentiate different types of polymers of plastic, and except when they are black plastic, which is based on hyperspectral imaging near infrared and in the short wave infrared region. I'm sorry for the pronunciation of this long technique. Anyway, it is based on the imaging analysis and also chemometrics, and is capable to differentiate the different types of plastic, which will serve also for, in addition to plastic sorting, has been developed a prototype, which also is expected to help to monitor the system for waste material. This, you can consult these results on Horizon Results platform. And For the pretreatments, in this first action of the project, we also tested how we could increase the biodegradability of the plastics after treatment with exposure to ultraviolet, with chemical, thermochemical treatments, or other treatments like EV radiation. At the end of this quite a big study, we found out that the best pretreatment to enhance the potential biodegradability, not only for the modification, because of the modification of the oxidization of the plastic, because of the cost and environmental analysis, was the exposition to ultraviolet during long time, 500 or to 750 hours, which is quite a long time. Yeah. But also reducing the particle sizes at one side is not that good because we have to release in the environment, we are releasing microplastic, but for some treatments, reducing particle size shows to be important, especially for the use of the microorganisms and the potential modification of the microorganisms. So going to the next step of the action of the project, that was the selection and the upscale the production of the biotech tools. We come from the basis of the idea that the polyester and especially polyethylene are very difficult to degrade, mainly because they are carbon to carbon, they are not oxidized, which is important. In fact, the microorganisms usually, the ones that are capable to degrade, have to oxidize it. For example, with enzymes like alkane hydroxylase and this. But there is some earlier reports that say that insects, especially insects, and also earthworms, and also so many references when we started the project, are capable of transform or partially degrade the plastics, and this action has been is known to be related not only to the enzymes that those organisms produce, but also to the microbiome that they have in the gut, in the digestive. So, in fact, it is expected that when the earthworms and also the insects feed on the plastic, the microbiome get adapted to this. So this is a good environment to search for microorganisms or new enzymes that could do something with the plastic. But the degradation is only partial in this case. So we say, okay, we can help this process. If we add microorganisms, for example, it is better, as it is in nature, to have a mixture of microorganisms, which is a consortium, because they help each other. It's what happens in the environment. So let's work with consortia. And which activity is good for us to build up the consortia or to search for this activity? If we look at the nature, there are complex molecules like lignin, which is very, very recalcitrant and very difficult to degrade. So let's go for ligninase, and also for other enzymes that we are known to degrade or to break down the bonds in the plastics. But we can, or our ambition was to go a step beyond this, is let's go for enzymes, what we call at the beginning, seems to be very pretentious, enzymes better than nature. I mean, it's new enzymes that could have better performance than the ones that are naturally produced by the microorganisms or other systems. So here we consider microbial consortia, insect, earthworms, and the combination of all of them is expected to improve or maximize the transformation yields of the plastic, or degradation will allow the treatment of misset plastic because they are in the waste fluxes. It's a mixture of different plastic, and maybe to convert the fossil fuel plastic into biodegradable counterparts in a single step. For example, insects feeding from plastic and then giving the chitin that is in the exoskeleton, together with all the other members of this equation. So we focus on those, the macrobial consortia. For the insects, we have been working with the gill mealworm, Tenebrio molitor, the glitter wax moth, Galeria melonella, and also some study with the Hermetia illustrans, which is the black soldier fly, and also Hephaestia pueniella, which is not in the picture, but it's another new insect that we have been working with. And for the earthworms, one of them has been Eisenia fetida or Andre, which is involved in the vermicomposting of the, remember that scenario with organic matter or organic fraction of the municipal solid waste contaminated with plastic. And Lumbricus terrestris, which is the earthworm, which is in the soil, that will be helpful for the bioremediation of the soil. So this is a summary of what we obtained in this first step, which is the selection of the biotech tools. We selected 4 microbial consortia that then, as I will mention in 2 or 3 slides, was used, one of them or 2 of them, for the soil and for the compost and for the or for treating the plastic from the outside, directly the plastic that are in those fluxes. This is why we call them the exo part of the equation, right? And we also obtained 2 novel enzymes that were synthesized, which is cutinase, 2 cutinase, which are fused with peptidase. And this is, we also call exo because it's the other part that we use to treat the plastic in those environments. The earthworm selected, as I mentioned, were Eisenia fetida that were used for this fluxer with plastic, with organic matter, Lumbricus terrestris with plastic contaminating soil, and the insects, Tenebrio, Hephaestia, and Galeria, the test we did to select and to monitor in the case of earthworms and insects was to see if they survive and if they have some activities with the plastic. And also for each of those insects and earthworms, we analyzed the microbiome and we isolated microorganisms from the digestive after they were fed with plastic, because in this those microorganisms are supposed to be adapted or help the insect and the earthworm to digest the plastic. And this is what we call endo, are microorganisms that has been isolated from the digestive or earthworms and the insects. And then we feed the insects and the earthworms again with those microorganisms to fortify them, like a probiotics. I don't know if you know this concept, like a yogurt, right? So this is what we do. We call endo because they were supposed to be working from the inside of the insect or the earthworm. Now I will show you just a few results, maybe not much information, but just a few of them because we have testing and selecting in different environments and in different conditions. This is one of the microbial consortia that later we use for the treatment of the plastic from the exo, from the outside of the organisms. We tested or analyzed or isolated or studied bacteria and fungi that has been isolated from soil, compost, from marine water, and also that has been obtained by enrichment method, by adapting in lab conditions. The enzymatic activities exhibited by those microorganisms has been used to design, to build up the consortia, the combination of 2 or 3 different microorganisms. Mostly are composed by Bacillus, Pseudomonas alternaria, and Fusarium, which are some of the microorganisms that we have used for those consortia. And the plastic degradation has been analyzed only for thanks to the capacity of the microorganism, the consortia, to grow from the plastic. We have tested all the plastic that we selected at the beginning, the 4 types of plastic in recycle and also in virgin form. When the microorganisms are capable to grow from the plastic as the sole carbon source with no other nutrients, we consider that this consortium may be successful. And together with this, we also analyzed the plastic weight loss. In fact, we obtained weight loss of LDP between 12 and 14%, which is quite a lot, but in 30 days, quite a long time as well. And for here you have 2 of the consortium that then we work with in different experiments. One of them is consortium 2, which is composed by Bacillus, Fusarium, and Alternaria, 2 fungi and 1 bacteria, and the other C4, which is composed by Bacillus and Pseudomonas, and both are capable to grow from the different plastic, quite a high level, and also they are capable, some of them are capable to modify the plastic. Here you have a picture of the LLDPA, FTIR, and the oxidization they provide. As for the novel enzymes, which is the other part, EXO, they were produced novel enzymatic chimeras with the fusing the cutinase with PKAS, and those were capable to reduce the weight of the polyesterase. They have a polyesterase activity. They reduce the weight of polycaprolactone in 10 days. Up to 97%, and it is interesting that they are also heat stable, which is a quality which is good for the enzymes, for the polymer degradation, and they are also capable to degrade LDPA with weight loss of 1 to 1.7 in 10 days. Also, there were novel enzyme synthesis, thanks to in silico screening and in vitro testing. 2 of them, the 3, well, they were analyzing different microorganisms originally isolated from fresh marine environment, and 2 of them, DH3 and DH5, which were also stable, heat stable at 50 degrees. Were also capable to degrade polycaprolactone and LLDP in 10 days. And in addition, there were improvements on the activity of those microorganisms or the activity of those enzymes by providing one important quality or property for the microorganisms that produce them, which is the capability to colonize and to form biofilms. The first step in the degradation of the plastic is the colonization, and the production of biofilms, the polysaccharides, is very important for the microorganisms to attach to the plastic. So our partner from Brunel will find another way to improve the activity of those enzymes. This, together with the microorganisms that I have shown right in the last slide, are also included in the Horizon results platform, and you can consult them anytime. As for the insects, are the other part of the selection that we need to first we needed to have to carry on with the next step of the project. First, the insects were tested in the different plastic using a standard diet for the insect together with plastic in different forms, as a powder and also as a film. Well, the insect prefer the standard diet, so they doesn't like much the plastic, but it was possible, as you've seen in the pictures and at the bottom of the slide. They were biting the films you see with the arrow, and well, they more or less better survive in standard diet and with plastic, but well, the results were not that bad. Well, some of the insects were cannibal, and they eat each other, because if there is no just plastic, also prefer to eat each other. But well, not bad results. And some of those microorganisms that survive, insects that survive in the plastic, we took the digestive and isolated. In addition to analyze the microbiome in general, we isolated microorganisms that were, you see in the petri dishes, the microorganisms that were present in the digestive of the insect that were fed in the plastic with plastic, but not in the insect that were not fed, were fed with a standard diet. And those microorganisms, singular microorganisms, were isolated and identified, and the enzymatic activity analyzed, and here we selected at least 2 or 3 microorganisms for each insect that was going to be used as a probiotics to improve to fortify their digestive and to make them stronger. And then we also have to analyze if they were compatible. I mean, if we feed the insect, if they were going to survive. And for example, in this case, which I think is for Galleria melonella, we selected Bacillus and Rhodotorula, and we saw that the presence of the added microorganisms help maintain the viability of the insect that is fed with plastic. In the case of earthworms, we did more or less the same in the case of Eisenia fetida and Lumbricus terrestris. All of this is in the lab, at lab condition, right? Eisenia was capable to growth in organic flusses polluted with plastic, but show a lot of higher susceptibility to the presence of polyethylene in comparison to the other polymers. In the case of Lumbricus terrestris, survive well with plastic, but the weight of the Lumbricus terrestris was reduced in the case when it grows with polyethylene or polyethylene terephthalate. In this case, we also isolated from the digestive the microorganisms, and we selected the unique cultivable microbiota, and that was going to be used as an endo component of the system to enrich the degradative microbiota. And at lab scale, we show that the presence of those microorganisms also help, but in this case, it's endo and exo together help to allows higher survival of these organisms when they are together with, when they are together with what? With the microorganisms. So we scale up the production of the microorganisms and the enzymes, and also we designed how was going the microorganisms to be fit to the with the insect and with the earthworms, and how to design the process in general. We designed different formulation and scale up the production, and the next step was going to be the upscaling and the to transfer this to the real scale. First, we tested the operational condition, and you know that the mineralization is the key step. If we consider biodegradation, is what to tell us if there is degradation, true degradation or not. For this, our partners analyze in liquid form the consortia, the effect of the consortia, and also the earthworms that were fortified with the microorganisms, for example. And well, at the beginning, they found that there was a release of CO2, and there was a mineralization provided by one of the consortia, but then this was not replicated. After a long time incubation, there was not so good as we expected at the beginning, and even in complex, or even using plastic treated with enzyme, or using plastic treated with ultraviolet, we were unable to find significant progress after long time incubation. So this was not so nice. But anyway, I will tell you shortly, I'm sorry very much, that the different scenario, because this is why we translated the different biotech tools to the close to real conditions. We have 3 scenarios. For the direct disposal, we reached the insects. In this case, we selected an embryo monitor for the upscaling of the transformation. For the organic waste polluted with plastic, we used the vermicompost, composting and then vermicomposting using the earthworms, together with the endo and exo organisms and also enzymes, and the agricultural soil polluted with plastic, Lumbricus terrestris, for the in situ soil bioremediation with the Lumbricus. Here you have the results for the ex situ degradation by insect rating with the embryo monitor. Well, unfortunately, we did this with mixed plastic and also with LDPE. Unfortunately, you see, we feed the insect with agar, where the microorganisms were inside, and they eat this agar with the microorganisms. We did not see clear visual evidence of plastic degradation, but also evidence of cannibalism, because they were eating each one. For the organic waste polluted with plastic, there was the situ degradation with composting and vermicomposting, fair composting, and then vermicomposting. We add the earthworms with the microorganisms. At the end of second phase, which is the vermicompost, worms were still alive, which is good, but the plastic was still present. But we showed that the plastic size was bigger than at the beginning. Maybe this is something good, but consider that this is a complex system where it is very difficult to study the plastic that remain after the treatment. I will not go deep inside of this. This is the case of the in situ bioremediation of soil with the Lumbricus terrestris that you can see there. They were fed also with the microorganisms, endomicroorganisms, and we found that the presence of the microorganisms alleviate the stress of the Lumbricus terrestris. And also there were 2 devices, one device and another result that we obtained here, the monitoring system that can differentiate the different kind of plastic, and also a model to discriminate, a predictive models that can monitor the biodegradation. Finally, the products, one of them was a biofertilizer that was made with insect frass, with the faces of the insect together with what they feed on, mixed with vermicompost, and we obtain a biofertilizer, very promising biofertilizer. you see here also in the Horizon Result platform. And well, this is the blending and then pelleting, and those 8 different formulations were obtained that can be used in the conventional farming and organic farming that has been validated on lettuce and cabbage. And the other products are insect byproducts like chitin and insect residues. Chitin is a product that can be obtained not only from insects but also from crustaceans and other organisms, and has antimicrobial and antioxidant properties. They has been extracted from the insects, and the case of chitin and chitosan through a green process that use e-bean to produce mulching films with enhanced properties and also coatings for active packaging. And also from insect residues, after the proteinization has been obtained filler that for the production of pots for the agriculture. This is the film. And first the extraction, here is the extraction with e-bean, extraction of chitin and transformation to chitosan, thanks to this system, the e-bean. And this is another result that comes from the project. And this chitin and chitosan is being in fact used as a coating for food packaging, specifically for chicken breast. And this is some of the pods that has been produced from the insects remainings after protein extraction. And finally, for the sustainability impacts, the 3 scenario has been compared with the reference. I will not go because I have no time, and also this is only partially performed, so I will not go through this. We also have made a logistic approach for the large scale implementation that is being analyzed in a region in Tuscany with companies or rural area, and that take the waste to a single waste treatment company. This is more or less the conclusion. From using tools we have developed from all of our process, it is true that this should be further investigated. The conventional plastic are hard to degrade and remain after most of the treatments. One key may be reducing crystallinity to increase biodegradability. The process with the selected microorganisms, airborne or insects, produce some mild signal of the oxidation or reduction in thermal stability of plastic, but this analytical is still ongoing. Insect frass-based fertilizer, a promising alternative to conventional fertilizer, and byproducts from insects are used for bioplastic production with promising results. I would like to thank to external advisory board that helped us in different meetings, and also, of course, to the partners of the project. Here are they, and here are the different sessions. You see, at first, after the 3 first sessions were online, and then we could join together. Thank you very much. I'm sorry for the long speech. Thank you.
Thank you, Maria. Are there any questions to Maria?
Thanks. So I'm not correct. So I'm a bit concerned about the temperature. And when you isolate the microbe from microbiome, I'm not sure whether you use temperature or higher temperature, which is only used to the so when you reintroduce the bacteria, you have the temperature inside the worm.
Yeah.
So my question is, which is the difference of temperature between the cultivation condition of these microbes compared to the temperature that this microbe will work?
We usually work with thank you for the question. We usually work with temperature around 30. I don't know, Ronan, you for fresh marine water, 30 degrees. So I think it's not that far from what you will find in, I don't know, which is the temperature inside of the airborns. I don't know, they are not homeotherms. They must have different, lower temperature than us. We are homeotherms. So I guess that they are 20 degrees maybe, so they are not that restrictive. But what we wanted to find is the enzyme that were capable to degrade the plastic at high temperature. Yeah. Except for the compost, which is different, yeah.
Any other question? No, then I think we are all hungry. Lunch is waiting outside for us. And we come back at 2 p.m. for the interactive session. So looking forward to it. Thank you.
One of the big possible solutions, because the enzymes are really quite good in degrading the polyester. And then, so we discuss how we can make this process more efficient. And so we found out that engineering is key. So we need to optimize the wild type enzymes to really adapt it to an industrial process. Depends on at which pH and which temperature, substrate load, product release. So all these things, product inhibition, play a major role. And with engineering, we can solve these problems. And then we also found out that we are now on the edge of real big transformation. So we believe that computational approaches will become more and more powerful. And the research we may need years for, maybe we can break it down in some weeks or months using AI technology. And the good thing is it will learn. So that means we make the algorithm even better. So therefore we think one key is also to engineer the enzymes with these computational approaches. And yeah, then also where to find these enzymes. I think it's good to know which kind of enzymes are out there in the environment. So making bioprospecting is also key to understand more about these enzymes. And then yeah, look on what we can still optimize. For instance, was that very interesting, like polyamide, polyamides, polyurethanes. Still here we have a lot of homework to do, let's say, because here yeah, lacking these potent enzymes. And then of course, yeah, the question was, can we combine the microbe and the enzyme? And we found out that for recycling, it's not working with the microbe. It's too complex. The downstream is getting hell. We need a single enzyme for recycling, what we think. And then for bioremediation, real chance to use whole cell systems, because there the enzyme alone is inefficient. So we need then the host where we have more time on that task. Yeah. So maybe I give the mic to the second group.
Thanks, Christian. Yeah, so from the discussions we had, we share a lot of the conclusions of what Christian already mentioned regarding yeah, the diversity of plastics and yeah, the different potential that enzymes has depending on what stream or what plastic you are looking at. And some specific ones that we talked about were, for instance, PLA. For which yeah, there is potential, since it's a polyester, to use enzymes to depolymerize it. But there are also certain challenges with respect to material performance and material sorting and things like that. So yeah, a more general theme that emerged from our discussions is that maybe developing an enzymatic recycling process should go hand in hand with redesigning or designing your plastic material. as was also shown in one of the projects presented today, that it can be quite successful, and maybe even that can be considered for current streams that are difficult to recycle, such as PET, PE, multilayer materials, to redesign them in a way that makes their enzymatic recycling even more effective. Yeah, the rest of our conclusions were very similar to what was already talked about by Christian. So if you have anything to add, Anna?
Yeah, regarding the bioprospecting areas, maybe we can also looking for fungi. Is also a relevant microorganism that maybe can look for some relevant enzymes, and also for thermophilic environments or other extremophilic environments, as for instance acidophilic environments. Maybe we can also find some interesting enzymes there. With relevant characteristics for depending the process, the enzymatic recycling process that we want to achieve. So another thing that is very relevant here is the thermostability trait of enzymes looking for, and also that the conversion rates is not only just a critical aspect itself, but also we have to consider other aspects, as for instance the techno-economical relevant aspects, and also the industrial relevant conditions, not only the efficient conversion rate. So comparing this mechanical enzymatic mechanical process is not so straightforward, because we have to focus on other aspects. And the direct evolution is something most promising tool to enhance all of these enzymes, especially in this industrial relevant environment. And the other conclusions are quite similar, they have previously mentioned. So that's all for our side.
Thank you. Thanks a lot. I took some notes, so please just check afterwards and add whatever I've gotten, because we take a picture of this. And I'd also like to collect your envelopes then, so that you leave it here on the table, and then we can prepare a big summary for each of us. The next group is the recycling group, the blue ones. So please come here. Wants to start?
Recycling, right?
Recycling.
Yeah. Hello, everyone. Actually, I took my notes already on that board. It's okay. I remember them. What?
Should we bring it?
Yeah, sure. Why not? My handwriting is not very good. So the first topic was how optimistic are we in terms of bringing the technology to the markets? What are the main obstacles in terms of enzymatic recycling, of course? And the common sentiment that I detected is that, sure, the technology seems very nice at lab scale, but what about the scale up? So far, the biggest test that have been reported in peer-reviewed papers reach 100, 150, maybe 200 liters scale. And we know that for bringing the technology to the market, we need thousands of liters. So this is yet to be seen and reported with numbers. But in terms of obstacles to achieve commercialization of this technology, there is for sure the economic side. We know from our analysis that there is definitely a premium compared to mechanically recycled PET. Will the market be willing to pay this premium? And how much is too much? And in terms of main cost drivers, it seems that the enzyme cost, enzyme loading, as well as the cost of the PET feedstock itself seems pretty relevant and important to be considered. There was also some interesting discussion related to biodegradable plastics. I think the general idea that I detected is there is some negativity in comparison to reuse and recycling. And this is justified by the fact that there were some, let's say, shocking findings recently in the scientific work where several biodegradable plastic or so-called biodegradable plastic were tested for biodegradability. And the results were that they actually stayed there for many years. They were not as biodegradable as we thought they were. So there is the question, what is biodegradability? Is it true biodegradability? And also, is the performance of this biodegradable plastic good enough for them to be used in mainstream applications? Think about food packaging, barrier properties, mechanical strength. So these are all things to be considered. Then there was one interesting point about this idea of embedding the enzyme directly in the plastic, so that once the packaging reach the end of life, you just treat the waste with heat. And since the enzyme is already in the plastic, we should somehow melt or depolymerize. But I think most of the people agree that this is science fiction, because I think there were some trials.
It's done now. It's existing.
It's existing.
There's a colleague present here that knows much more about it.
Okay, but there were some questions related to the fact about, is the enzyme still active once you extrude it at above 100 degrees Celsius? And what about the cost? Because we need a lot of enzyme that is used only once, is not recovered after its job is done, unlike, for example, the polymerization in a reactor where you can recover some of the enzyme. So there was some skepticism for this technology. Yes, and these were the main points, I think. I mean, there is much more, but these were the most interesting points.
Okay. Okay. Hello. So I will start with the hardest of the enzymatic recycling. So basically, we were talking about the low TRL and the lab scale rates, and also the problem with the separation of the waste. For that, well, as you know, it is difficult to separate when there are black plastics and also with the dyes. However, I know that sooner it's going to be possible to separate it between multi-layers, okay? Then also this is applicable mainly to polyol films and aluminum and other ones. And also it's important to know that it's contaminated by heavy metals. That's another issue regarding the sorting. Also, it's another hurdle when recovering the resin or the product. Okay, and then what are the cost drivers? The cost drivers. So when obtaining monomers, that's expensive. So basically, the suggestion here is that the EU should give incentives to the companies. For example, also removing taxes to the companies that are getting products obtained by recycling. Okay, and then regarding the identification and sorting of the materials, we were talking about how we can improve that, given that there are limitations such as the black plastics and the multi-layers that I was talking before. So applying digital traceability, like for example, adding some markers into the plastics. So this is a way of labeling them, so they can be separated and traceable afterwards. And applying any pre-treatment process at the beginning, such as cleaning step, also could help in improving the separation. Then we were also talking about the importance of process manufacturing regarding the enzymes at the extrusion compounding processes. So for this, well, different suggestions were given, such as the need of adding a softener extruder, non-degrading additives for the enzymes, and basically the problem here is the temperature. So it's very difficult to find a thermostable enzyme, even though engineering solutions at the devices have been implemented and should be further implemented as well. There is the need to protect them through maybe this is magic, but an encapsulation procedure. And also it would be also interesting to work with the small enzymes that could reactivate faster compared to the larger ones. It's also another solution that we were seeing here is that applying any sensing technology in line by monitoring such reactions would control the situation by, for example, also controlling externally by a digital twin or something that can work and close the loop in real time. And then, well, just finalize that for the enzymatic recycling, the big challenge for the cost and the lack of efficiency is related also to the difficulties in the upscaling.
Okay, thanks. Next one.
Thank you. Well, we discussed also, well, regarding the gap between lab scale and the real world. In some cases, we have seen that there's maybe, or it's little than we think, or maybe there's no gap at all. But in other kind of like PET, but maybe in other kinds of materials like polyethylene, the degradation is much more difficult. So maybe the nature of the plastic is one of the main obstacles for enzymatic recycles, because those materials that imitate or present some bonds present in nature are more prone to be enzymatically degraded. And then also we discussed the collecting of the material, the cross-contamination, although the enzymes present the advantage that they can work on so-called contaminated materials or materials in a mixture. Also, we also discussed that in the case of textiles, in the PET of textiles, it would be beneficial as well. So I would say sorting is important as well. And then about the cost drivers, we discussed the enzyme production, but mainly the downstream purification of monomers for the recovery is one of the main cost drivers in this process. Lastly, from my side, we discussed that the pretreatment of the material is key in these recycling processes regarding the surface area, the surface area ratio, and also the crystallinity of the material. So the more amorphous of the material through extrusion processes, the easier for the enzyme would be to be degraded. If you want to add. Yeah, we work together a little bit, so not much to add. Only, yeah, collection is important, that you have homogeneous streams. Can be done now with good labeling systems like a water marking, which is now existing and should be a solution for any kind of plastic packaging. So we have to reduce contamination. That can be an important factor in the system. Indeed, enzymatic recycling, we think it's still a little bit expensive, but it's really specific. You have good monomers and you reduce then the downstream process. That's good. And also with a good pre-treatment, you can ensure that your
enzymatic process goes much faster. If you have a high surface volume ratio, or if you can reduce crystallinity, it will improve the enzymatic recycling process. So we believe that in a circular world, I believe also that apart from mechanical recycling, also enzymatic and chemical recycling can play a role, and enzymatic recycling might be more environmental friendly even than the chemical recycling. So that might be an interesting approach, and I hear just now that they are really considering already building plants which are offering enzymatic recycling as a key aspect. And I'll be a little more bit in this meeting, I suppose. I have also wanted to say on biodegradation. Our company is doing biodegradation tests, so that's important. You mentioned something about biodegradable materials that were not biodegradation showing in the environment. It's important to know that biodegradation is really related to the environment. You have to select to test biodegradation in the environment you want to have it. Probably they use also a lot of compostable materials like PLA. This PLA is really easily industrial compostable, but it's not degrading in soil and aquatic environment because it needs a kind of temperature for the hydrolysis and to start biodegradation. So we really have to consider in detail what did they test, did they test in a good manner. Normally a product that has been showing biodegradation in good lab conditions, it should degrade also in the environment. Of course, it's depending on moisture, microbial activity.
I think the main difference between industrial compostability and home compostability.
That's it.
So, material that can degrade really if it's liquid environment quickly in a matter of months, or it requires specific conditions in an industrial composting facility.
So that's there.
I think there's a lot of confusion.
Yeah, and that's again, labeling is important also to show where it's working there, and that people are throwing it in a good direction. We believe that actually you have a bio-based material and it's biodegradable or compostable, it can enrich the current plastics that are now occurring because most of the plastics, like PLA, you can also mechanically recycling. You can easily chemical recycling, or enzymatic recycling, they are even more easily to degrade because they have these ester bonds. So they can offer many more applications in degradation than the current ones. But of course, we have to look at each application for that. And in fact, it's already done that enzymes are already added to plastics to promote the degradation, like for PLA, in more mild conditions that are degrading at lower temperatures. That's my contribution.
Thank you. And then the last topic, upcycling.
We leave it here, no?
Do we leave it here?
Yes, please leave it here. Yeah.
That would be good.
Well, you can do it.
Otherwise, I will ask. Thank you. Who wants to start? I think you have been one big green group, and then we separated you into two.
Yeah.
So I give you.
Yeah. Thank you. So in the upcycling groups, so we address basically two main questions. On one side, we discuss about the characteristic of the mixtures that can be recovered after enzymatic degradation and that we want to reuse to make materials afterwards. And on the other side, we discussed about the specification and requirements in order to use this matter in order to make plastics and to revalorize this monomer mixtures. So for the first question, the main points that were raised is for the moment we obtain complex mixtures that with quite low yields and quite a lot of variability depending on the plastics we are degrading. So this raised a lot of questions regarding how to valorize it. We discussed about two possible ways of working. First way would be to say, okay, we should valorize these mixtures without additional treatments or more efforts, because it's the straightforward, cost efficient, but it can bring a lot of variability in the final materials because we're going to have different starting materials, so different mixtures, and so different final materials. The other approach is more on, we need to understand more these mixtures, so develop analytical methods in order to really understand what is in the mixtures, implement separation protocols, treatment protocols in order to finally get out the pure components out of these mixtures that can be substituted to the conventional petrol-based chemicals in order to make plastics. Okay, so this is on the what we obtain part. So what we're gonna do with it, questions. Is first, we need purity. We need to know the molecules we are working with. So to have assessment of the purity, of the functionality, avoid mixtures. The volume was also an important question. In order to address certain markets, we need some volume. So we need these processes to be consistent in terms of productivity. So basically, compared to the first discussion, we need to increase yields of these processes. There was also a discussion about what are the plastics at the moment that can be considered for this kind of degradation and revalorization routes. Because as we have seen this morning, it's working quite well for PET, it's working for some unsaturated polyester resins, but we also figured out this morning that for PE or for other thermoset resin, it's not working. So maybe there is a point to address here to really identify what are the plastics that should be considered for this kind of recycling routes, and which are the ones that cannot be addressed by that. Yes. And this is also implying a question of sorting, because if we decide to address this recycling route for some plastics, it means that we have to handle the sorting of this plastic as a starting point, and which is also a great challenge, even more when we see that we have multi-layers, composite materials and everything. So it can be really challenging. So in terms of challenge, as I said, understand the mixture, make in place separation protocol, also assess the energy and process variability, because we also discussed about the upscaling of these processes. Okay, it's working well at lab scale, but when we're going to increase the quantities in order to make more production, one of the questions is what about the energy we're going to put into these processes in order to produce enough materials to address markets? Question of solvents and all this stuff, and so go way beyond. Go way beyond in the LCA analysis in order to ensure that what we are doing is in the good direction. And finally, we discuss about the market, and I was speaking about what plastic we should consider for these recycling methods. We should also think about what market can be addressed with this plastic we're going to produce. And what came out is we need to focus at first on low quantity markets, because for the moment the yields and productivity are quite low. High added values, market that can accept high prices, because for the moment this supply chain, I would say, is not ready yet. So we need this kind of markets, and also low performance materials, because as we have seen for the moment, the monomer that can come out this kind of processes are quite low performances, and so as a starting market, we think that it can be the best option.
So we just go to us, because we have a lot of points in common point. So the first point was about the monomers, and we confirm that we has to use the monomers need the purity to use various building blocks for different applications, not necessarily the one from which the polymers are coming. The enzymes seems to be very specific, so in some situations can even work on mixed plastic. The main issue is when we have mix of PET and polyurethane, so it will be very valuable to have a salting between the PET and the polyurethane, which are often together, especially in carpets, shoes, and other applications. So sorting for these two polymers will be very good. And if we can have this also quite separated plastic waste, the enzymes looks to be quite good for getting back to the monomers, which then are building block, so can be used as a starting point. And so difficulty. Another question was if enzymes can be reused, so can be recovered and reused, because this should lower the cost. Because at the end, cost and market acceptance are the main limit to look for an industrialization, because the process can be industrialized, but then we have to go. The industry has to be competitive in the market, so cost and and market acceptance are the main challenge to be met for being for getting on after is a business plan for the continuous of the project. So then we have valorization via biomass, which results for the different project and the different action. In recover, we propose the valorization of insect biomass remaining as filler, because it's mainly calcium carbonate and some chitin. So either extraction of a chitin, because it's very valuable, or use as a dry millet powder as a filler to replace calcium carbonate. So not high value application, low level applications such as the pots and so on, even because we like the idea to put back an environment biomass which appertain to that environment. So putting some exoskeleton on insect in the field is something natural. By putting our calcium carbonate coming from our process, maybe less natural in some way. So we like the idea to put back something which is already in some way in that environment from the beginning. And then there was a question about the biofertilizer, because we valorize the FRAS as biofertilizer, and this is working very well, but we don't know if it is the standard, the normative, for them towards the using of the insect FRAS and as a biofertilizer. In Italy, it's allowed, by our country now, so we don't have it yet in European clear idea. Well, this will be very good, very nice, very valuable, because it was one of the best results we had in the COVID. So in general, knowing if we can use this biofertilizer, and this can be accepted by Europe. So this will be very important, even because we know that the topic of the biofertilizer is quite hot with the agriculture sector in recent time too. So I think these were most of the things added to the one we already addressed. Yeah, so that's it. So we see a lot of opportunities of valorization and upcycling from the three projects, independently and together. So let's see if we can go on.
Thank you. Very nice words for the end of the discussion.
Now we will move to the final speaker. Let me leave one microphone, otherwise. So today we will learn more about the black fly soldier. The company Enzycle is a special. Oh my God, it's black. Sorry. The company Enzycle is specialized in this organism, and they use it as a biodigestion technology. We are happy to welcome Maria Machado from Enzycle, who will tell us more, and maybe we find a good way to connect enzymatic recycling approaches with this special insect. The floor is yours.
Thank you. Hello, everyone. So, as presented, I'm Maria Machado. I'm from Enzycle, Portugal, and we specialized on insect rearing of black soldier fly larvae. So I'll talk a bit about this insect and how it played out in Recover as some solutions and innovations, and its role on biofertilizers and some processes and products. So black soldier fly is an insect that's originally from Colombia, the tropic region. Nowadays, it's a bit spread out even from all over the world, or the tropic area, and also some temperate zones. And this insect is really interesting because of its larvae, which is really rich in protein and fat. So its content, it's really interesting to use for feed. So it's getting famous by the day as an alternative sustainable feed source. Regarding its life cycle, it's an holometabolic insect, which means that the adult insect is different from its juvenile stage. In this case, we'll have the eggs that hatch for a couple of days in the really good conditions, and then we'll have the larvae that is our star here. The larvae grows for about a couple of weeks, and then the pupal stage is when the metamorphosis happens and the fly erupts after that. This insect can complete 4 to 6 cycles a year in a temperate zone. In the tropics, it can do a lot of more cycles because it's really good conditions there. And it's interesting because this larva can eat up to 0.5 grams per day. So it's a really voracious eater of organic matter and all sorts of organic matter. That's why it is so interesting to use it as a feed source. Also, not least interesting, the adults have vestigial mouth parts, so they don't bite, and it's not known to be a vector of any disease. So it can reassure us that it's safe to use and to use it in a commercial and a super large scale because it won't affect us in any part of our immune system, as other flies may do, as we know. And actually, it's really a nice insect for rearing because the fly is not really bothering you like domestic flies, which are always on top of you and biting and everything. So it's a really good insect to use for commercial and industrial use. So at Enzycle, we use this approach where we take vegetable byproducts from agriculture industry and also food industry, like processing food industry. And we create a substrate that we then inoculate with juvenile larvae. They stay on the substrate for a couple of days eating it. And after a couple of weeks, sorry. And after that period, we sieve them and we get 2 fractions. The first one, which is the larvae, we can then process into protein and oil, and we can add this to feed. This feed can be used on fish, pet food, and also pigs and poultry. And the other fraction, which is the frass, which already has been talked here about today, is an interesting biofertilizer that can be applied to the fields and also food production. So in Recover, we try to use these species to plastic degradation. Unfortunately, we weren't that successful because due to the mouth parts, this larvae has more of a sucker mouth parts. They were able to eat the plastic. Although there are some literature that says that when you cover the plastic in mollusks, they are able to eat them, but we haven't tried that because that was not the focus, covering the mollusks part. So that's why here we have represented a tenebrio larvae that was integrated in project to the plastic degradation bit of the work package. So, but the BSF larvae was really able to contribute to the soil recovery through the biofertilizers application in the soil, and also to contributing to bioplastic work package with chitin-based polymers with their exoskeleton transformation of chitin to chitosan, and then develop a lot of products. So here we have an example of a work package of Recover. On the left here, we have a picture of one of the trials we did. And this is a really interesting byproduct of this production because it helps the soil gain more structure, and it helps to enhance the water retention, and it really promotes the development of microorganisms, so it improves the biodiversity fauna of the soil. It also, because the frost as bits of the insect sheds along the larvae growth. It also promotes the immune system development of the plants. So when they are attacked by plagues, they can be a bit more resilient and fight through better. And we have fortified plants with this usage of biofertilizers. They can be used either in powder or pelletized, as you can see. And we have a really good outcome with productions using this type of frost. But also in other work packages, we tried to apply this, since they weren't able to eat plastics, we tried to use the better what we could do with them. So we used a mixture of municipal waste, which was a bit of everything. It was really smelly and uncomfortable to be around. So we received this municipal waste from a partner, a couple dozens of this, and we separated for different crates, rearing crates, and we inoculate them with juvenile larvae. And for about a couple of weeks, we left them there. It was curious. Then around 2 or 3 days after, the smell almost completely disappeared. So it really showed us the capacity of transforming this matter from the larvae. And after a couple of weeks, we had this result. This was after sieving these 4 crates. And for the first 2, the bottom 2, we have the smaller particle portion of the frost, which is the organic matter all decomposed by the larvae. And the other 2 are the bigger particles of the decomposition, being the 4th one, the on top one, the mixture of all inorganic matter that the larvae was not able to digest. So this is not, well, it was not great that the larvae was not able to eat plastic, but they could eat the organic fraction. So they could be used in this case, where they can eat all the organic matter present in the substrate and then leave the inorganic part for your enzymes to transform. We also did, in another work package, we developed these pots, as Maria Jose already mentioned, and this is kitten-based pots and also with portions of the insect, but this is not really the only product that you can develop from kitten and chitosan. You can go as long as your imagination wants to, because it's a polymer. So I really believe that in the future we can see a lot more of these type of products in the industry. In the project, we developed these pots and some films and mulchings, and also I believe there are some filters already being developed, but not only focusing on kitten, because we cannot use these larvae that treated waste to use as feed, because it's not allowed because of safety reasons, obviously. But we can use the oil and the fats. So this oil has really good characteristics and can be used as in cosmetics, and also in the industry vision-wise, it can be used as lubricant or a biodiesel oil. So it can be one product with many different usages. That was it for me. Thank you very much for your attention. If you have any questions.
Thank you, Maria. Very interesting as well. Very different.
Yeah.
Is there any questions for Maria before we conclude? Yeah.
Thanks for the presentation. So I have one question related to the biofertilizer. So I assume to be used as a biofertilizer, you need to control the nitrogen composition or so, or probably you are going to change the rhythos, the composition of the plants compared to the normal traditional fertilizers. So do you have any information about this? How it changed or not?
Well, actually, the frass is really constant in its organic matter and nitrogen content, but it can have some fluctuations according to the substrate the larvae developed on. So if we have a more fruit waste, it will be with some characteristics, but mostly the minerals like calcium and phosphorus remain the same. So the variations are very little. I don't know if I answered you, but.
I mean, you compare additional fertilizer, chlorified fertilizer, to what extent change the microbiome in the soil once you are.
Oh, I'm not the best person to answer that. I'm not the expert on the biofertilizers, but we have seen from our trials that there is an increase in the microbiome regarding other organic substrates. So we do believe that it promotes the increase in number and diversity.
Maria, I just want to ask you again a couple of legislative questions. I don't know if you can answer, but firstly, I think that using food waste to feed the black soldier fly, that then you use that as a feedstock, is illegal still, isn't it, under EU law?
No, we're not able to use food waste, not yet at least. There are some projects regarding that safety matter. We can use, though, byproducts of the food transformation industry. Okay. So when you process the food for our frozen, we can use the byproducts of that production.
Okay, and the second question is, and I think Professor Cinelli mentioned this morning as well, is about the use of fertilizers. And within the fertilizer regulation, is that feedstock allowed?
Sorry,
the larva from the black soldier fly, is that, what do you call it, the frass, is that allowed as a feedstock to make fertilizers under the fertilizer regulation?
Yes, actually we are, we have, because the fertilizer legislation is a bit controlled from authorities for the country. So it's not clear for all European countries, and some of them has a more loose legislation than others. In Portugal, we are having some struggles, but it's not the fact that the larvae are fed on byproducts is not the problem. It is more the characteristics of the frass and the innovation of the product that is more scaring for the legislation, not as much the product in itself. I don't know if I was clear, but I thought it.
So in other words, it's allowed at an experimental stage, but it's probably not yet allowed at a market stage.
No, it is because the legislation is not really clear, because we are using nowadays like chicken poo and horse poo to has fertilizers. So this is not as different from those. The difference here is the treatment. Well, not actually different, because we are subjected from the same treatment as those are. So the frass pass through a thermal treatment, which we believe it's not helpful because it kills a bunch of microorganisms that would be helpful for the soil. So that is what we are fighting on the legislation, to try to change a bit the treatment so it can enhance its properties and not degrade them.
No more questions? No? Well,
thank you.
Maria. And thank you. Everyone is awake again. We had a very nice evening yesterday, so thanks to the organizers from the Bizente project who organized the dinner yesterday and also the city tour. I think it was a really nice occasion to get to know the city a little bit. We are now on the second day of our final event of Bizente Recover and Enzycle. And just one announcement at the beginning. Today, yesterday, 2 codes were left here. So if somebody misses his code, it should be over there. And also a notebook with some notes was left that's outside at the desk. So if you don't find your notebook, it might be there. While yesterday we focused a lot on the results and on the achievements of the project and also application fields, we will today look into policies, strategies, and also get to know other projects who are also working in this field of plastic recycling. And we will start today with Samuele Ambrosetti from the Bio-Based Industries Consortium. He is Innovation and Programming Manager in Brussels, and he can give us some insights into the big position and the cascading use of bio-based resources, including recycling.
Okay, thank you. Thank you very much for the introduction. Hello, everybody. Thanks for inviting me to this exciting event. I'm sorry I was not able to follow the first day of the event, but okay, I'm here for the second day, and I'm very much looking forward to the discussion and the debate and the presentations. I hope everybody can hear me well. Okay, and so yeah, thank you very much again for inviting me and for giving me the honor of sort of opening, the dances today. Yeah, just very briefly, I will introduce who we are. Can you see actually the screen? No, okay. Where do we need to go? Because if I move the arrows, it's moving for me, but not for you. Now it's moving. Great, fantastic. Okay. Well, no problem. I remember sort of what I put in slides. So, well, I see quite a few of our members actually in the room. It's always good to see familiar faces, but for those of you who don't know us yet, I represent the Bio-Based Industries Consortium. We are a Brussels-based organization. We have now almost 300 actually members. The original infographics is from January, and since January we gained almost 40 members, and it's growing. We are an industry-driven organization representing basically all the sectors of the bioeconomy, from the primary producers, agriculture, food, forest, aquaculture, marine, algae, microalgae, waste managers, processing industry, packaging, all the way to the chemical industry, of course, biotech, all the way down to the brand owners who are bringing the bio-based products to the market. And I say industry-driven because it's not only about industries, but also about everything going on in the research world that can be applied in industry. So one of our missions is bring together these two worlds. Well, we bring together many worlds actually. The original name of the partnership was actually Bridge, because we wanted to build bridges between sectors. And the reason we were born more than 10 years ago was to be the private side of the BBI, so Bio-Based Industries Joint Undertaking, which I think all of you are familiar with. We are still performing this role. The partnership now is called CBE, Circular Bio-Based Europe Joint Undertaking, and it's the first of our 4 pillars of activities that you see on the right-hand side of the slide. So we are still representing the industry in this wider sense that I meant in the joint undertaking. But with time we have evolved, let's say, our model to also perform other activities, and the second one is business, we call it, which means we try to create opportunities for our members, industry and non-industry, to get together, cooperate, develop new projects, new products. We have matchmaking events, networking events, etc. The third one, we call it finance, because of course, in order to bring research to the market, you need to have investment, and that's why we work with the more than 30 now banks and financial institutions, including the European Investment Bank, the European Circular Bioeconomy Fund, and in 2 weeks we are going to have an event for startups in Ghent, in which the startups can pitch in front of the investors and hopefully find, let's say, a good match. And the fourth pillar of our activities is what we call society, and this is what I will be focusing most today in my presentation. So under the society pillar, we, let's say, group all the activities that we do to promote the uptake of bioeconomy and bio-based solutions in our own lives, in our society. And this means both the work with the education institutions, from primary school all the way to postdoc, but also, and this maybe is more important for today, the work of advocacy with European institutions. So in this framework, what we do is we have this kind of privilege, let's say, position as being the private side of the joint undertaking. So we have a day-by-day connection with the European Commission, especially the GRTD, but also the other DGs. And so we try to, of course, let's say, build sort of mutual understanding of what the challenges are for the bio-based industries, what the opportunities are, and how the policymakers could support the bioeconomy in Europe. So I will start with the quite well-known, I guess, diagram, butterfly diagram, just to remark where bio-based products and bio-based solutions can fit in this picture. And it's pretty much everywhere, of course, in the left side, because it's everything that is related to renewable feedstock and resources. But let's not forget the right side, because we apply biotechnology in recycling, for instance, and somebody in the room is very knowledgeable about this. So we, of course, as bio-based, operators, we are very much focused on biomass feedstock and renewable feedstock. But let's not forget that even if we want to achieve as much as possible the use of renewable and bio-based feedstock, we will always need, as a society, some virgin fossil raw material. But if we are able to recycle it, of course, it's even better. And let's say, from our point of view, when we talk about bio-based loop, we're talking about this. We are talking about, as much as possible, using a cascading approach, as much as possible trapping carbon in durable products, so that while the feedstock is renewed, the carbon stays there, and we have even a negative, possibly, carbon value chain. And when we talk about, okay, we don't think it's taboo to talk about primary biomass feedstock, because of course we need to take care. We don't want to take away, let's say, land from food, for instance, crops. We don't want to, let's say, promote deforestation. On the contrary, we want to promote sustainable forest management, sustainable agriculture. But we think that the bio-based industry, so let's say exploiting biomass for something, for non-traditional uses beyond food and beyond, let's say, pulp and paper or construction timber, is a key element to provide diversification also of income for the operators and to build synergies between the sectors. And at the end of life of the bio-based products, you have multiple options. So it's, of course, reuse, it's, of course, recycling, but then many things can also be composted, or many things can return to the ground and bring nutrients back to the environment in the form either of compost, of biochar, or even by biodegrading in the environment if the application foresees it. So we see multiple advantages of bio-based products and solutions. And then this question always comes up, yes, but what about the biomass gap? Because we say we want to promote additional uses for biomass, but we don't have enough biomass, because we have this big biomass gap. Yes, but let's look at the bottom of the picture. So most of the biomass used today is for energy and fuels. So what happens if we reduce or even get rid of the energy part? The biomass gap reverses. We have more biomass than we need. And a substantial part of it comes from recycling and bio-waste. So this is something that we need to tap into. And of course, we will never probably get to the picture on the right, because it's overly ambitious to completely decouple biomass from energy. But still, if we were able to reduce that use and promote the use for materials and chemicals with higher added value and also lower emissions, probably, of course, that could help. And talking about emissions, also here, we think we have an edge as bioeconomy, because we can basically improve the situation in all the three scopes, both upstream and downstream. And it's not only a matter of improving, but if we combine the storage of carbon in durable products the cascading steps and feedback loops back into the system and carbon capture and use, we can even have negative carbon emission from our value chains. But one big problem that we see currently is that the PEF and the ERCL methodologies are not accounting for this. So this is also something that we are working on, especially with DG Grow. Let's say we, as a part of this advocacy and what I call the society activities, we are doing many things. One that I didn't mention actually, which is quite important, is that you remember in the beginning I said we're trying to building bridges between different sectors, but it's not only a matter of sectors, but also a matter of countries, a matter of geography. No, Europe is a continent made of many different countries. Not all of them have the same, let's say, kind of bio-based activities, the same kind of investment, the same kind of technology readiness, but all of them hold promise, all of them have opportunities. And so what we are trying to do with various actions is to stimulate the cooperation, especially regarding the so-called widening countries, so especially the Central and Eastern part of Europe. I think just yesterday we published a country report on the potential of Romania in terms of bio-based activities, which is very interesting, not only in terms of agricultural biomass, because of course there's a lot of agriculture there and a lot of forestry, but also in terms of knowledge, because there are some really, really good researchers over there, and maybe not everybody in the other part of Europe is aware. So I invite you to read this kind of publication. We have it for Poland, we have it for Romania, we have it for the Baltic countries. Hungary will be the next one later this year. But then what we also are doing towards the European institutions is trying to create this sort of awareness of the potential of bioeconomy on one side, let's say, provide a solution for multiple different problems, but also of the hurdles and of the obstacles that we encounter, which sometimes are not really, I mean, some of them are technical, and okay, we can solve them. We have the research projects to do that, but some of them are of legislative or policy nature, and then, okay, they're not really fundamental. They can be, overcome if everybody is willing. So what we did is we had a series of workshops last year with civil society actors, with European institutions, with people from industry, people from research, to talk about some different themes, and the outcomes of these workshops were collected in our trend reports that you can download, in our manifesto in view of the European elections, in which we try to set forth some of what we believe should be priorities for the new Commission, whatever the color will be, because I think bioeconomy, sustainability, strategic autonomy of Europe in terms of resources is something that is related to everybody. It's not depending from the political force in power. And the last thing, the very hot news, of course, you're all familiar with the Biotech and Biomanufacturing Initiative, just came out a couple of weeks ago. And let's say, in preparation of this, of course, we, as many other associations, provided our input, and we are quite happy that many of our points were taken up. I think the initiative is quite promising in terms of fostering not only the development of biotechnology, which is fundamental, but also the use of renewable circular bio-based feedstock, which is equally important. And the two things together, I think, can really unlock a lot of potential for Europe as a continent and as a society. Okay, so you can see here a bit of our highlights. One thing that we think Europe is really good at is financing research, maybe not so good at financing scale-up and access to market. For a number of reasons. Some of them are related to the maybe mindset of the European investors as compared to Americans, for instance, or from, I don't know, Singapore, which is really betting on it. Something more related to legislative hurdles. We know that getting permits in Europe is a bit more a longer thing, and getting a new product approved, for instance, for a novel food, is very long. And okay, the precautionary principle, of course, is something really good for our health, etc. And we should not, relent on it, but also we should maybe make things a bit easier for innovations to actually produce an impact. And so we are setting forth this request to speed up and ramp up the scaling capabilities of European innovation. Another thing that is debated, of course, as I mentioned before, is the biomass. Have we got enough biomass? Where does the biomass come from? Can we afford to produce more biomass while we also want to have more sustainability and we don't want to take away land from the environment? And of course, there are a bit of different priorities to balance. I think that if we took away a little bit of land that is used for animal feed, for instance, we could unlock a huge potential for the bio-based industry. But okay, this is my personal opinion. But still, we have so much to do with the residues, with residual biomass, with bio-waste. By the way, we will have a study on biomass availability coming up, I think, next month, and another one on bio-waste generation and management coming up in June. So we are trying to provide also to the policymakers the data so that they can have the real picture in front of them, because sometimes it's not easy to get this kind of data, and you all know about this. So once we have the data in front of us, we can substantiate, of course, these positions and these decisions. And then, of course, the overarching goal is we shouldn't talk about decarbonizing, I guess, but we should talk about defossilizing our industries, because we will always need carbon for materials, for food, for everything. But if this is renewable carbon and not virgin fossil carbon, of course, it's a different game, and we should be playing that game. And then I think I will stop here, because I guess my time is up, by giving you some of the next events and highlights, both in the CBE partnership and for ourselves. So the call for proposals of CBE 2024 is open right now. It will close in September, and there will be an info day on the 23rd of April in Brussels, which is a great also chance for networking. So I invite everybody to join this public event. It will also be streamed online. So, okay, if you cannot travel, you can also follow online. We will have the day after this startup event that I mentioned before with investors. And then we have the biomass availability and the bio-waste studies coming up, and a few other publications maybe in the second part of the year. And as you can see, the CBE topics of this year are pretty interesting, I would say, also budget-wise, because we have 213 million of budget overall. Many topics spacing, over different themes. So I invite you to go to cbe.europa.eu and take a look at the work program, because there are many interesting opportunities for everybody. Thank you.
Thanks a lot, Samuele. Are there any questions?
Yes. I should sit again.
Yes, so thanks. Additionally, I agree with you that the topics is really the topic of this event. I have a question about this biomass.
Yes.
When you are discussing about or reaching the market, something at the end going to the market also. I'm wondering the scale you should reach or is recommended.
Well, that's the $1 million question, isn't it? No, so in the CBE we are financing different levels. So from lab scale TRL 5 all the way to TRL 8 flagship level, with the innovation action TRL 6, 7 in the middle. And the idea is basically, each value chain, each application has their own scale that can be considered commercial. And for some applications, you have like a direct fossil counterpart, so you have to go to massive plants because you have to have economies of scale. Otherwise, you will always lose the price battle. Then, okay, if price is the only parameter, then I'm really afraid it will be difficult to have a a fair playing field. But for for specialties or for ingredients that you can basically only produce by bio-based means, then maybe you can have a smaller scale, or you can have different kind of like decentralized concepts working in synergy. So, I mean, yeah, that's not an easy question to answer. What I think is important is to pass this message that bioeconomy is no longer a risky business. I mean, it's something that investors can look at as a reliable market, and they can invest in it also at scale. Of course, it's still relatively new, some of it, not all of it, and so that's why we need this kind of initiatives. I mean, maybe they don't need the same kind of initiative in the petrochemical industry because it's established since 100 years ago. You know?
Hi, very interesting talk. You mentioned something very important. I think it's the supply and demand of biomass is crucial.
Yep.
As if there is a gap. Plan to provide maybe some reports or some information regarding what is the biomass that is currently unexploited, and it's currently available, but nobody's taking advantage of that, because I think that's one of the main hurdles of the industry, that people would like to use biomass, maybe second generation, especially second generation biomass that doesn't depend on fertilizer or agricultural land,
yep.
But they don't know where to look for. Also because often this biomass is collected, there is not a center that you can reach out to and easily get your hands to. So do you plan to maybe make a report or a study that could help companies to understand where to look for second generation biomass, and also available at a reasonable price that makes the unit economics?
Yeah. So there are multiple actually levels that you can tackle this. For sure, what we are doing now, I mean, we have this study ongoing and it will be published pretty soon, I guess, on biomass availability, and that will be looking at the big numbers, the big statistics. How much biomass we have? I think it will be reasonable to assume something at European and national level. But then the problem with biomass is also that you have very local value chains, because of course it's perishable, most of it. And so the granularity of data that you need is very fine. So this is really one of the big hurdles. The other is what you mentioned, the logistics. Basically, not all of these possible sources of biomass are even mapped or even known or even tracked. Then we have the problem that we don't want to deplete the soil. So whatever needs to be left on field, it's better to be left on field. No, we need to keep that kind of balance. So this is I think one of the main hurdles. But we do have some kinds of biomass that are separately collected: municipal solid waste, urban wastewater, the green residues from gardening and pruning. They are separately, the wood, they are separately collected already. They have their own logistics. Maybe we can think about shifting some of it from their current use, which is mostly energy, and make something different out of it. That would be, I think, the lower hanging fruit. It's not trivial at all. But then the second thing would be to go for these really unexploited and unexplored sources. But this, I think, will need some maybe concerted effort from a European level, but then it will need to happen at local level.
Thanks for the questions. I think we have to move on. Thank you, Sophia.
Thank you.
Our next speaker is David Newman, who is chairman of the European Bioeconomy Bureau, and we are very happy that he followed our invitation. His CV includes some very impressive stations, such as executive director of British Italy, leader of the Italian Bioplastics Association. He was president of the International Solid Waste Association in Vienna, and he founded and leads the Bio-Based and Biodegradable Association in the UK. So let's ask him about his opinion to fight plastic pollution and climate change with novel technologies.
Good morning, everybody. And again, like Samuele, I thank you very much for the invitation, the hospitality here. Fantastic. All conferences should be held in Madrid. I am going to try to tell you the history of the world in 15 minutes. It's a little bit difficult to do, but just to give you a brief introduction to the EBB, we do policy work, and we do policy work within European projects. We've been doing this now for going back to 2016. So if you are looking to bid for funds under the CBEJU and you need policy analysis, well, I hope ours is as good as anybody's and we're available to talk to you. I'm going to try to run through these questions here, try to link them up and understand where we are, and try to take your attention away for a moment from carbon and try to take your attention into toxicity. As we all know, we've got a lot of plastics out there. We all know where they come from. I think sometimes we're a little bit surprised that actually it comes from so many sources. And when you look at the different sources where plastics come from, you understand why it's very difficult to recycle plastics. And it's very difficult to recycle plastics which are clothing. And we're seeing what a disaster fast fashion is creating globally. It's very difficult to recycle plastics which are in agriculture because, of course, they're dirty. They're soiled by soil, by pesticides, by fertilizers. And so it's no surprise that we have tens and tens of millions of tons of plastics which we just don't know how to handle. And that's leaving aside packaging, where actually we are doing something, and not particularly brilliantly, but we are trying to handle it. The impacts of plastic pollution are grave, and I think we completely underestimate them. 80% of all the rainwater now contains microplastics and toxic chemicals, which come from plastics. And our air quality in almost every part of the world, I think bar the Antarctic, is beyond WHO safety levels for 2.5 PM 2.5 particles. All fish are toxic. And we can see, and I don't know if anyone saw recently in Florida, a video of fish spinning on themselves and then laying over and dying. And this, when we look at whales and dolphins, the levels of PCB in their brains are, well, huge, 50 times more than causes brain damage in us. And when we look at us, we look at the booming cancer rates that there are amongst young people. And I see many, many young people in this room. And at the same time, the falling human fertility rates, especially male fertility rates. And what this has caused, all this pollution, this toxicity has caused, is a plateauing of life expectancy. You know, over the last 50 years since the Second World War, we've been used to every year life expectancy improving. It's plateaued. It's plateaued. And this is because of the toxic load. So you ask the question, it's a little bit rhetorical, of course, will we be able to procreate in a few years' time? It's not so rhetorical, actually. And microplastic pollution raining down, you know, soils and is everywhere. Recently, I read about the baobab trees in Africa, which are all slowly dying because of the rain of toxic micro pollutants on them. And does anybody know what a coffee pod is? Well, a coffee pod is not a coffee pod. A coffee pod is the world's largest carbon reserve. And we've reduced that carbon reserve by 50% over the last 25, 30, 50 years by poisoning it through microplastics, chemicals, sewage, etc. And there would be potentially enough coffee pods were they allowed to live in a clean ocean to more or less absorb most of the climate change emission gases we're putting out. And here's a link where you can find a lot more about the role of microorganisms in the ocean. And the pollution coming out through our river streams, these are two examples, the Amazon and the Mississippi River stream, which are bringing down pesticides and plastics and fertilizers and chemicals, hormones, sewage, etc., into our oceans, create these huge great areas which are essentially dead zones. So this is all not good news, ladies and gentlemen, is it? We look at the oceans. Why the oceans? Because the oceans are actually 70% of the planet's surface. We forget about them. They generate 50% of our oxygen. They absorb about a third of our CO2 emissions. And the copepods and the life and the phytoplankton in the oceans take up and capture 90% of the excess heat we are generating. But if you look at the colourful test tube, you can see that ocean acidity caused by CO2 emissions themselves and by toxicity from the chemical industry, from the plastics industry above all, is actually rapidly increasing the acidity of our oceans. Now, you might not think that 8.2 to 7.95 is a big deal. Actually, it's an 18% increase in acidity over the last 40 years, and it's accelerating. So what does all this mean? Well, Copernicus just published last week the data for March 2024, which shows that the monthly global surface air temperature anomaly is a constant increase over the last 10 months, the first time that we've ever measured such a long increase in global temperatures continually above median. So it's a continual anomaly. It's no longer an anomaly, it's a norm. So let's look at some solutions. I think, and you can take this with a pinch of salt, this is my personal opinion, that we probably can't do very much in the short term to reduce carbon dioxide emissions. We are on a trend where the fossil fuel industry is continuing to expand, plastic production is continuing to expand, and the locked-in emissions we already have will see that curve continually upwards over the next years. But what we can do, and we can do quite rapidly, is decrease the amount of pollution we're putting into our oceans to help improve the sequestration of that carbon. And how? And we're talking about plastic today, so I'm only going to focus on solutions regarding plastics. There are several points which are, for us policy bonks, quite interesting. There's the UNEP ANC process, which is going on. As you know, the next meeting is in Montreal in a couple of weeks' time. It's a very, very hard process to try to cap or limit or reduce plastic pollution. The big producing companies are just going to tell you to go to hell. They're not going to buy it. So it looks like we're going to double plastic production by 2050 and not reduce it. But thanks to Europe and thanks to the research which has gone on, we do have compostable and soil biodegradable plastics. Samuele mentioned them before. China is leading the investments into these plastics. It's still very small, just 2% roughly. But hey, we've got them. Why are we not using them more? Above all, I think we have to pay for very high quality collection systems. It's great being in Europe because almost all of our waste is collected, but 65% of the world's population have no professionally managed waste services. And literally, you can walk around the towns of anywhere in the third world and you just see people with lorries just dumping them in the river, dumping the waste in the river. It's normal. 80% of sewage enters into our water system globally without treatment. Now, this is just a question of money. All the technologies are there. We have them. We're lucky. We use them every day. We need $250 billion a year forever to invest in these systems and to stop pollution going into our environment. Now, you've talked here about enzymatic reactions and enzymatic degradation. I'm no expert, so please don't ask me to comment. But a note of caution, we should not risk that researching into such solutions pushes back the need to implement other solutions more quickly. For example, the compostable and soil biodegradable plastics, and for example, waste management systems, and collection of waste. As you yourself said yesterday, you're a decade away at least from any sort of industrial scale implementation. So these are, in summary, things we need to do: waste management, less harmful, more benign chemicals. We have to ban some of those chemicals outright. PFAS. Why are we still talking about PFAS? And your technologies on enzymes. So we have to go from fossil fuel plastics to biodegradable, bio-based plastics. Why aren't we doing this? In fact, the whole EU legislative structure seems to be actually actively opposed to this transition, which is quite extraordinary, considering that we invented here in Europe these plastics. We have to go towards zero waste. I've never been a fan of the term zero waste. It's sort of, I don't know, coming from an NGO background, I like to be practical about the things we can do, and I don't think zero waste is a practical objective. However, it gives a sense of direction. And we've got to go from landfilling and incineration, which damage our soils, air, and water, to recycling, to composting, refill systems. We're going to have to change the way in which we live. And I know it's awkward, I know it's difficult, I know it's a burden, particularly for families with children and such like. But there's really no choice. But we need some policies. I was quite surprised yesterday to note that none of the projects had any policy analysis, any legislative analysis. And maybe that was because you didn't want to speak about them yesterday, or you wanted to speak about the science. But if you want such things as enzymatic solutions to plastic to come to market, then you probably are going to need some policy and legislation to help drive that. So, firstly, zero emissions. Let's forget and not focus. Forget that we focus totally on CO2. It's not enough. We have to focus on all emissions. So, for example, water, we have to look at the nitrate directive if that's going to be revised this year. We have to look at zero toxicity. I said I don't like the phrase zero because it's impractical, but it's too late to talk about a bit toxic. We have to talk about no toxicity. Now, the Commission and Parliament just caved in to the chemical industry a few weeks ago and to pressure from farmers to stop them using very poisonous chemicals in our food systems. This needs to be pushed back in the next legislation. Zero sewage to rivers and to seas, not just less, zero. Actually, we're still pushing out into our systems here in the EU, where we have all this money, all this wealth. 10% of our population still is not linked to sewage systems. And recycling, everyone wants to recycle, my God. But unless we make recycling the economic go-to alternative, it's never going to work. Now, you can't recycle plastics in the EU profitably because recycled plastics and virgin plastics are coming in from the Far East at a much lower price. So why are we fooling ourselves? So to get plastics recycling and material recovery up, we need to tax virgin materials and we need to subsidize recycled and recovered materials. By the way, plastics recycling in the EU, you all quoted yesterday 35%. That's not true. That's 35% collected for recycling. What actually comes out of a recycling plant is anybody's guess, and how much goes to Turkey, Malaysia, and Indonesia is still anybody's guess, perhaps 15%. And finally, let's break down those barriers to using these bio-based and biodegradable materials and textiles in plastics themselves, farming, fishing, fertilizers, pesticides. We have all the technologies, they're all there now. And yet we're poisoning ourselves by continuing business as usual. We are involved as EPB in a project with some of the other people here in the room. I can see ATIP and ENCO. Forgive me if I've forgotten anybody else. We're working on this project, and in this project we're trying to work out these scenarios of rural bioeconomy and how they can contribute to this industrial transition, making good the resource chains, the waste chains which we talked about that are available in the rural economy. So if you're interested in following this development, please go to the Brilliant website and join us and sign up for the newsletters. And of course, we will be having meetings like this in the future. So to conclude, we're on the cusp, or we could be if we wanted to be, on the cusp of a new industrial revolution. Let's be quite honest, the chemical industry in Europe's got no future the way it's going. It's just so much cheaper and so much more convenient to do chemicals elsewhere, where you have less environmental controls, where you have lower labor costs, where you have lower energy costs. So if we can't do cheap, we can do good. And green chemistry is the way to go for the European chemical industry. We've suddenly found out when Russia invaded Ukraine that, hang on, we're importing all our fertilizers from there, but we have all these natural fertilizers. We're just not valorizing. We're not collecting. We're not treating properly. We're not putting back to soil, as men have done for millennia. The Ellen MacArthur Foundation discovered some years ago that we're only recycling 2% of our organic waste globally. 98% of them just finish in the open environment, dumped, causing methane emissions as well as pollution. Plastics, they're all made now in Asia, in the Middle East, America. We were leaders in bioplastic. We've allowed that leadership to fall away, and now we have to recover it before it's too late. What are we all going to be, ladies and gentlemen, in 20 years' time? Waiters? Cooks? Bus drivers for rich tourists coming here from India and China? Or are we going to have an industry here in our own country which can compete, which can supply our needs? We've got to look at changing farming methods. This drives me absolutely crazy. 1/3 of our EU budget is spent on the common agricultural policy. 1/3, 60 billion euro a year. And yet farmers are upset. They're angry. They're protesting on the street. And yet we cover them in money. We give 1/3 of our budget for 4% of our workforce. Can someone explain to me quite why? So we need to have this deal with farming over the next few years and start a new green revolution, which is producing food which is healthy for us, and soils and water and air quality, which is good for us. We are actually incentivizing farmers to pollute us. It's really, really ridiculous. And I'm afraid the Green Deal has become a little bit of a grey deal. And we need to make sure that when we go to the polls in June and elect MEPs in our country, that we should be asking them these questions. And Samuel put some of those questions out there. What are, in the manifesto, what are we supporting? Are you going to ask an MP to defend nature, improve agricultural and health outcomes, or are you going to allow him to continue to vote for business as usual and pollution? That's your choice. And sadly, I don't vote. I live in London. Thank you for listening.
Thanks a lot. I have questions today. First.
Thank you. Great presentation. Great presentation. I like the idea of farming green revolution. So, I have a lot of experience with the local farmer, the people, animals, take a lot of plastic for many different plastic labs, plastic animal reproduction, and then they take the plastic, just put in somewhere, burn, spray. So I think local farmers, people working with animals, with farm, need a solution, not at the big scale, because we are thinking about plastic at a big scale, but the local scale. So somewhere the project or the policy work will be done in the local scale also. Because I'm pretty sure the farmer would like to have the solution there to deal with the design, renting, destroy. So I think this farming or animal green revolution.
Okay. I have no need to comment then.
Thank you, Mr. Newman. I think you had a very important presentation. So you mentioned the fact that during the presentations yesterday, there were no points related to policies. So I think I would like to give my contribution. From our economic analysis, it seems that for enzymatic recycling, it would be crucial to create an economic incentive directed towards the waste feedstock, because that represents a big part of the unit economics of recycled PET from enzymatic recycling. So it would be important maybe to create a tax break or a carbon tax that is applied on fossil plastics, and then this money is redirected to recyclers that then can have waste feedstock at a very cheap price, and then so this is my opinion. This is crucial. And then a second point, I was quite surprised when you said that all the technologies are here for the green transition. Well, I kind of disagree with that. I work in a venture capital fund and invest in the bioeconomy, and I look particularly at packaging and construction. And for packaging, for example, what we notice is that there is a lack of solutions for certain applications. For example, think about food packaging. New materials, sustainable material, and also especially bioplastics, they lack the performance needed to prevent, for example, the vapor or the oxygen to enter into the packaging and make the food perish. So if we would switch to this material, we would have much more food waste because the performance is just not the same. So I think we are still a bit far from that point of view, and we still need a lot of research and innovation to make sure that we have the materials that we need to make this my two.
Thank you. Well, on the first point, I think Samueli touched on it. We need a biomass strategy. Okay, we need a biomass strategy urgently to create that level playing field between burning biomass and creating materials from biomass. So I know this doesn't quite answer your question, but yes, we need to figure that out. And in terms of answering your question around enzymes and incentives, of course, it's quite a hard sell to go to the plastics industry and tax them. And you will find that your MEPs, as in the packaging debate, will be chased into the toilets with pieces of paper saying, you have to vote for this, you have to vote for this. This happened in Brussels just a few weeks ago with lobbyists for the packaging industry pressuring MEPs even when they were going to the bathroom. So, you know, the oil industry, the gas industry, the plastics industry are very, very tough industries to work against. Regarding your compostable and your bio-based and bioplastic materials, I don't quite agree with you. We have today, for example, even coffee is now packed in 100% compostable packaging. You know, ground coffee is a particularly difficult thing because it expands, you know, and so that's why you have that little filter on the coffee pack, because it has to let the air out. That's all done today with compostable materials. So, you know, I think that there's small scale still, but their innovation has virtually overcome all the barriers, almost all the barriers. Maybe with a little bit of your capital from your VCs, we'd get there.
Thanks.
Thank you very much for the presentation. My name is Substitute from the Spanish Research Council. So my question is regarding toxicity. So you were talking about plastics, but I'm wondering about toxicity of not bioplastic, but additives that we are using to make bioplastic. So as far as I know, so they are similar, that the additives that are being added to the plastic, but then we are using composting system. So it means that these additives are being transferred to the land, and the certification that we have now, the system for the certification, from my point of view, are not really measuring the long-term effect of these additives. So what is being considering now from the point of view of the policies, because I think that we have to make it right now. So we can't wait to find the effect in the environment. As well, it's the same in the open environment application. So what about these policies?
Yeah, I mean, just because something is biodegradable or bioplastic doesn't mean it should have a free pass in terms of toxicity. It has to have the same, it has to have as good or same toxicity levels or lower toxicity levels than anything that we have on the market today. I think that you will find Oh dear, you will find that everything is measured, of course, against food contact. Everything is measured against the 13432, and therefore earthworms, etc. I ran the Compost Association for 15 years in Italy, and we tested for toxicity on all the bioplastics and all the compost produced using bioplastics. We never found anything that was potentially damaging, but that doesn't mean it's not there.
Talking about the long term.
Yeah, exactly.
With the certification we're having.
Short term, sure.
It's not an impact, but what about regulation in the
Yeah, I mean, there's green chemistry has got to step up its game. We have to start producing materials and chemicals which are toxic.
I think that this is one thing that is missing.
Yeah. Yeah. Okay.
Okay, thank you very much.
Thank you.
I think we are running out of time. Thanks. Next speaker is not the one in the agenda, but he's replacing Fernando. It's Roberto Orcayara. We practice the pronunciation. He's from the Eurofunding office, and he will also tell us something about strategies and policies.
Sorry. That's a PDF. That's not the other one. Thank you. Well, thank you very much. I'm sorry Fernando is not here because he knows far more of this than me, but I will do my best. I cannot do more. So, what I would like to talk. Okay. I will talk about first what is the picture of the production of bioplastic in Europe, and then we will go hardcore into policies. We'll face some challenges and some opportunities. I will try to do quickly because we are already late in the agenda. To start with, look at this picture of the European bioplastic, that they are fantastic making this analysis. They are 1.9 million tons in bioplastic production in 2022, but in the EU, this only 0.3 millions. This gap is something that it has been described later, and I will sort of mention after that, is because many bioplastic are coming from East Europe because they are producing cheaper than us. So this is some challenge that we have to face in the future. The European policies, again, the European bioplastic is fantastic in this. Has been working in the past and is still working, and the idea is to adopt a policy framework for sourcing, labeling, and the use of bioplastic, as well as the use of biodegradable and compostable plastics to contribute to sustainable plastic economies. So we have from the European plastic strategy to go to the Green Deal, which is the cornerstone of these policies, and from there we can have other policies like the European Biodiversity, the Circular Economy Action Plan, and the idea is to clarify where the bio-based plastics can provide environmental benefits, under which conditions, for which applications, while holding them to the same strict standards as any other materials. The idea is not to build bioplastic by bioplastic, but bioplastic that clearly has some environmental benefits, while having the same use and hopefully in the future the same cost, which this is another challenge that we will have to face. So what has done the policies with bioplastic? So what the Roman did for us? Let's start with the Green Deal. The bio-based plastics can contribute in the Green Deal policies, like increasing the EU climate ambitions from 2030 to 2050, to mobilizing the industry for clean and circular economy, as well as going towards zero pollution ambition. I know it is I'm agree with the latest point to see zero waste and zero pollution is a little bit over the top, but try to minimize as much. Of course, there is the sustainable development, the UN Sustainable Development Goals, that the European bioplastic goes straight into the air, for example, in affordable clean energy, industry innovation, infrastructure, sustainable cities, etc. Also, the waste framework direction and the landfill directive, particularly because sooner or later we have to recycle that, and this will be an issue in the future. We will talk about that later. Of course, the bioeconomy strategy action plan, contribution in bioeconomy strategy objectives like reducing dependence of renewable resources, mitigating and adapting climate change, and also industrial policy strategy, managing fast inclusive transformations, champion global competitiveness, etc. There are more, much more, like the single-use plastic directive, like the packaging for food, which it is tricky, like the European strategy for plastics, the Circular Plastic Alliance that have an objective of 10 million tons recycled plastics, the sustainable, the EU strategy for sustainable circular in textiles. In the textile is so important in plastic because the low cost clothes are generating a lot of waste in plastics. The soil strategy. I mean, there are many policies that are now on and ensure that will be modified in the future. My previous speaker has mentioned several suggestions that I fully agree in where the policies could come. So what are the challenges we have faced? First, cost competitiveness. At the moment, we are still not competing with the cost of petrol-based plastics. Like, for example, if you compare polyurethane, polypropylene, etc., with polyhydroxyalkanoate like PHH, you can see the difference. This is something that research and fundings have to work together and trying to close that gap at some point. The limited production capacity. I saw in the first picture that the difference between the global bioplastic production and the European is so much. It's because, let's face it, the petrochemical industry finds easier and cheaper to produce petrochemical plastics. The low market share also, and just to put, and we have to consider several. I have several data that we have to take into consideration. In Europe, any person will consume around 150 kilos per person. That means that the plastic production in Europe it's around 57.2 millions of tons. That was the data of 2011. And the post-consumer recycled plastics attributed to bio-based plastic, well, this is something for the future, I haven't noticed that. It was around 2.3%, while only 9% of the plastic ever produced has been recycling, and most important, 12% of the plastic has been incinerated. Regulation hurdles. I mean, the policies are there to help, but sometimes they are confused. And we need to consider all these regulation problems, like the single-use directive, the recycling plastic for food and for food-containing materials, all the certification. There are regulatory issues that when we are producing plastic, we have to consider. Sustainable claims in environmental performance, the Sustainable Products Initiative, etc. It's not in here, but probably the most damaging harden from the regulation point of view, and it is something that I will mention later, is the fact that there is not homologation between the national policies between one country to another. So, for example, one waste or one biomass can be used in one country for one area, but in another is not able to use. We need this regulatory harmonization in order that if we produce a bioplastic from this biomass can be used in Spain and all the way up to Finland, passing through all the middle of European Commission or European Union. So this is probably one of the most important issues that we have to face. Another thing that we have to face is our environmental impact. Again, bioplastic, when you produce bioplastic, you also have an environmental impact, and you have to consider. And there is, for example, just put one example. If the European Commission, the Green Deal, etc., work, and we start using bio-based material to make bio-based plastics, there will be a moment with all these PHAs, these polyhydroxyalkanoates, etc., that we will need to consider recycling these bioplastics. This is something that at the moment we have not faced, because we know very well how to do PET, how to recycle PET, how to recycle polyethylene, etc. But nobody has thought in how to recycling polyhydroxyalkanate. First, because it is mixed with many other things, we will have to separate it, etc., and then compose it and do it again. So this problem is something that has already been looked upon, and the European Commission for one side and the industry, the Bio-based Industry Consortium for another side, thought on that, and last year they launched this call of recycling bio-based plastics. So this is one example in which policies, industry, and a European Commission are working together to facing these challenges. So what are the main challenges we have to face? First, the techno-economical viability production process. We need a realistic and feasible business plans for the bio-based plastics. We need innovative plans for recovery, reuse, and recycling fossil waste plastics and complex polymers. For example, composite, which is a nightmare, and is another elephant in the room that is about to explode. For example, the blades of the windmills that are about to end their life now, and there is no way they think how to recycle these composite materials. Traceability of the circular economy. We need to demonstrate that this bioplastic is circular, and there must be some way to demonstrate it. Example, the green passport. Another example is in the next line, digitalization of digital economy. We need to keep improving digitalization. For example, in blockchain, there could be another way in which we can trace the circularity of any bioprocess, but also the incremental of artificial intelligence, the use of digital twins for production optimization, and then reducing this gap between the prices of bioplastic and plastics. For example, we need holistic life cycle analysis and life cycle costs, because in all the projects that we are doing now in the innovation action, we have an ex-ante life cycle analysis, and then we do a life cycle analysis at the end of the project, which is level 6. From there to the industry, fighting about these regularity things. It is important. If you want to buy in Spain, in Holland, in the Netherlands, sorry, in Finland, etc., you have to have the same rule for everyone at the regulatory level. Supporting for entrepreneurship. Not only as the incubation of a small and medium-sized enterprise, which is the accelerator is doing a fantastic job, the accelerator program I will mention like later, but also in the creation of new companies. And these new companies that try to survive a little bit better, not to become the little turtles that were born in the sun that most of all of them died. Try to be a little bit more protected with this new startup creating regarding biomass. And of course, public and private financial support. We still need money, come on, let's face it. So about facing the money, let's find funding opportunities in Horizon Europe. You can find opportunities everywhere. You can find horizontal, well, bottom-up opportunities. In, for example, if you are universities at TRL low, like European Research Council and Marie Curie's, And theoretical heights you can find at in the European Innovation Council, like the Accelerated Program, but also you can say the Pathfinder and the Transition. You also can have opportunities in the European innovation ecosystems. For example, the Eurostar program is part of that. And of course, in the Pillar 2. I mentioned the food, the cluster 6, which is the one that deals directly to bioplastic, but could be everywhere. For example, tomorrow there is a call on the health area that talking about bioprinting materials for renewable energy. This bioprinting material can be bioplastic scaffolds to grab their cells, for example. So bioplastic for health can be used. Bioplastic for space. So that can be everywhere. And of course, don't forget the circular base Europe, which is the ones. If you see in here example of past funding opportunities, they are everywhere. So this is examples of funding opportunities closed. I tried to find open ones, but well, there is the CV program. I didn't know that you were going to introduce it or not. So I'm sorry, Samuel. If not, I will introduce the topic for the CVs. But this is the past examples. The cluster 6 is closed. As you can see, most of them are CVs and cluster 6. But don't forget that there are other outside the European, the Horizon Europe, there is another opportunities that you may have not considered, like the LIFE program. Like there is this whole section of circular economy and quality of life, where they will find close to market projects, but innovative projects also, which integrate beyond the air, water, and waste and other areas, such as circular economy. And finally, and this is probably more for industry than probably big industries, something that many people may think, which is innovation funds. Those innovation funds are projects to develop first of a kind industries, highly innovative, with one requisite. They have to demonstrate with some particular indicator that you will reduce the CO2 emissions in your process. It is nice because it not only finance the OPEX, which is the everything related to the innovation, but also the CAPEX, the construction and building. And it can reach even up to 90% in CAPEX in some cases. This is everything I wanted to say. So from my side, thank you very much, and I open to questions. Yes, I haven't talked my book. That's true. I'm sorry. That's unforgivable. Yes, my book is Funding is a consultancy company. We work in helping people get the funds. So we are consultancy in European funds, but not only European funds, also national level, tax incentives, environmental. We also work in the PERTE in regional funds, etc. This is pretty much. I am involved in the European project department. We work the programs we set, so European Horizon Europe, but also Eurostar's accelerators and innovation funds that we actually are probably one of the few consultancies in company in Spain that got one innovation fund granted. Yes. Just to finish my book, our success rates across the whole European environment is around 40%. We are quite successful in accelerator programs. That's my book. Sorry.
Thank you, Roberto. I think we will make a very short bio break now.
Yeah, thank you, and thank you for the invitation and to present our project, which has a topic quite different to this event, but yeah, so it's a project in the topic of using enzyme to make consumer product greener. So I will show in the presentation which are our objective, the team, and also some synergies I see with the other partner involved in the three projects discussed here, and also for the future. So our project, the Future Enzyme project, is a project that meets the demands of the consumer. So consumer, industries, and also the planet. The consumer, because we are all concerned about our planet and our consumption choice. This for sure, we are all aware of this. Industries, because they are trying to make our product every time greener and greener through innovation, and also because the planet, because we know that our geosphere and biosphere are changing. So we need to reduce the CO2 emission, global warming, and so on. And we all know that with enzyme, this can be solved. So we can provide benefit to consumers, the industries, and also for the planet. And our project is targeting three sectors: detergent, textile, and cosmetic sector, that altogether make 40% of the market, the European market. They are highly consumed, so this is why we target. They are related to high carbon emission, and this is also why industries are trying to make innovation. And we all know that with enzyme, we can reduce this CO2 emission and chemical release to the environment and so on. And then the idea of Future Enzyme is taking enzymes, so we can go from natural enzymes in the environment to our house, to consumer. And there are many different challenges, because you can understand that the enzyme required by detergent, textile, and cosmetic are very different. The product require different temperature, different working composition, and so on. And then finding the enzyme is not easy. It's costly. We all know that actually, currently, we need about 30,000 euro to discover an enzyme. The screen itself is associated with a carbon footprint, so we need to optimize the process of identifying an enzyme, because we need computer power, person power, experimental power, and so on. We need at least about 15 months, so we are reducing this time. Even the production of the enzyme during the fermentation is also associated with carbon footprint, so we are lowering this carbon footprint by optimizing the production cost. And from lab to industry, a single we need to characterize more or less 100,000 enzymes to get one to the market, to this product. So we are trying to optimize this, and the objective of our project is just to establish and combine a series of technologies, bench and computing, to retrieve highly powerful, stable, and low-cost microbial enzyme with the aim to approach the complex reality and challenges of real consumer products. So the idea is to get 3 consumer products in the market, and then make greener. So these companies that are Henkel, Evonik, and Schuler can make greener the product already accepted by consumer to make greener. So it's a clear objective. So what we are doing is just we get a clear understanding of what the industry wants. So the characteristic of the product, so where the enzymes should work with, the temperature condition, the pH condition, the composition, and so on. And then with this knowledge, we take a lot of genetic resources from the environment, from public databases, and so on. So then we just screen by in silico bioprospecting, computed bioprospecting, and so on. So then we express all the enzyme, we characterize, we accumulated a lot of metadata. And with this metadata, then we feed computing. So we make predictive tools. So the idea is every time we get a sequence associated to an enzyme with a characteristic required by detergent or textile or cosmetics, so then the machine we will automatically predict whether this enzyme is good or not for our purpose. And then we select enzymes that are fitting to our product. So then we make a lot of engineering by in silico, artificial intelligence, and so on, and also bench. Also, we have protection strain, manufacturing process, also, both at the level of the enzyme and the product, and then we make pre-validation of our product. So I have a video summarizing that probably summarizes better than myself with a better English, I think. But it's just a 3 minutes video. So.
How to reverse this while keeping our way of life? Nature provides a solution. Microorganisms and the enzymes found in their genomes can be bioprospected and computationally analyzed to disclose those that can be of interest for green applications, bioeconomy, and circular economy. Enzymes are natural molecules that perform all the amazing reactions that allow life, so we can learn from them to pamper the earth. Come with us to see how we can help to make the world green in detergents, cosmetics, and textiles. In textile manufacturing, compounds such as spinning oils and dyes are complicated to remove from the final garment, needing a big amount of water and energy for its heating. Enzymes can remove those, avoiding such a high utilization of water and energy. Moreover, disposed garments can be treated with enzymes, so the material can be used again as raw or even be degraded. In laundry detergent formulation, the use of more efficient enzymes at low temperature and more stable enzymes at any storage conditions will improve laundry neatness while reducing energy and water consumption. And less chemicals for the formulation to be released to urban sewage. Hyaluronic acid is a commonly used compound in cosmetics due to its moisturizing properties. But its size is key. Enzymes can cut big hyaluronic acid molecules to a precise size, replacing the commonly needed processes that use high water amounts at elevated temperatures. When the hyaluronic acid molecule with the perfect size is applied on the skin, it has the highest penetration power in the dermis and avoids allergic reactions, while obtaining the best anti-aging results, moisturizing the skin and reducing wrinkles. In future enzyme, we aim at implementing our awesome enzymes in these everyday products and carrying them to the edge of biotechnology to be applied in green industrial processes.
So the video, so this summarizes the video. So here are all the panels involved in the project. I think it's more or less is summarized. So I will not give any experimental details, and then just I wanted to show here what the synergies I see that our project can contribute to the solution of bioplastics, and I think it was already mentioned a few times during the previous presentation, the problem of the textile. I think the European Commission is working in the transition pathway for the textile ecosystems. So this is here. And the question is that we are working not only in enzyme for removing these spinning oils and dyes from during the processing of plastic, but also for the degradation of textile based in polyester, and also with base of elastane, with this polyurethane. So the idea is just to just to give a number. So it's second a truck of clothes is thrown away or incinerated. So the idea is we are thinking that adding enzyme to the recycling process can result in saving about 5.5 kilograms of CO2 per kilogram of textile material. So if we extrapolate this to all the textile throwaways, so I think the numbers are quite high. And what we offer, and I think is a synergy, because currently very little polyester is recycled. Indeed, 99% of the recycled polyester is coming from PET from bottles. So I think it's a good point to start working in the recycling and reuse of polyester fabrics. And a synergy between our project and another project, and in the future call, will be to implement enzyme to degrade and reuse polyester textile. And especially in the area where we are working, or at least our industrial partner Schuler is working with, is sportwear industry. So this is a synergy I see from our project. And also another synergy is because in enzyme we develop, in future enzyme we develop a complete pipeline, bench and an artificial intelligence baseline, and also engineering pipeline, and also the production of enzyme. So we are able to identify enzyme from the environments, then engineer and produce. We have a lot of accumulated knowledge in different enzymes, including plastic degrading elastane and polyester degrading enzymes. So I think this accumulated knowledge can be a synergy to your product. And also we are aware about the policy, not only in the textile sector and plastic sector, but also in the enzyme itself. So we can make a synergy at the policy level, working in demonstrating the safety and sustainable use of enzyme, demonstrate how enzyme technology support the European Green Deal and circular economy and other areas. And also, which is really key currently, is recommend that enzyme should be exempt from the amendment of the REA regulation because the enzyme are thought to be respiratory sensitizer. So this is something that was not claimed today or during this event, but then now the European Commission is stressed this problem. And then we need to make a lot of policy work to really prove that the enzymes cannot be claimed as a respiratory sensitizer. So including for the recycling of plastic. So we can do a synergy on that. And then that's all. So this is just to acknowledge the team of Future Enzyme. This is our last event. And then if you want to see, we have a lot of for student comics and so on and videos. So feel free to go to the webpage and then see. And that's all. So if you have any question, yeah, so fine, okay, yeah.
Thanks a lot. Are there any questions?
Yeah. Yeah, thank you. Yeah, we cover, I will say, most of all types of extreme environment on Earth, all types. So deep sea water, mountain, polar sites, acidic, hypersaline, saline, but also most of the normal, a lot of marine sites. It's incredible that for plastic degradation, most of our best enzymes are coming from seawater. It's incredible. We are learning now that the best enzyme for hyaluronic acid degradation comes from salty water. So we try to accumulate metadata from which different kind of enzymes rich. So we know now that for plastic degradation, and mostly from seawater, from hyaluronic acid from saltern sites, and so on. But we target a lot, and also public databases. So because we feed our computers, but then the computer are just screening the public databases also. Yeah, and then there is a lot of concern about this Nagoya protocol. So any of our industrial partners, Henkel, Schuler, or Evonik, or other, are receiving an enzyme if we don't prove that this enzyme passed the criteria of Nagoya protocol. So we are aware that the company can use and market this enzyme and the process. So yeah, I think it's important to clarify the origin of the source of the enzyme. Yeah, so what we implement is just to clearly clarify that we have all the approval of the local government, and when the sample, so we record everything, when the sample was taken, with the person involved, how many partners were involved in the discovery, in the sample uptaking, discovery, production, and so on. So then just to clarify the royalties or patents and so on, but especially to have the local authorization that the sample was taken in a correct way and with approval. Without this, so we don't transfer any enzyme to a company, it's for sure. Mm-hmm. Yeah, thank you.
Thank you very much.
Mm-hmm.
Another question here. Thank you very much.
Yeah, thanks.
I have a question concerning your cosmetic application. So I saw that all your partners is Evonik, so I guess that you are using bacteria cellulose. So the point is, what are you adding to this application? And your small molecules of hyaluronic acid, but are you also associating enzymes to cosmetic? That is possible, or is this not approved? Or
yeah,
what are your approach if you mention? Is just producing the product that you would like to your application? Or
yeah, it's a good question, and perhaps I didn't clarify. So the hyaluronic acid cosmetic is just adding hyaluronic acid to the cosmetic, not enzyme. The problem is it's very large molecule. If it's too large, it can penetrate into the skin. But it's too small, then it's toxic to our body. So they need to cut the hyaluronic acid in the perfect size, and this perfect size is about 1-2 kilodalton. This is almost impossible. This is almost impossible. Almost, it's very, very, very difficult. And then what Evonik is trying is just to find an enzyme that produce a hydrolysis product of this size, and then integrate into the cosmetic, and then compare the anti-aging properties of a cosmetic with this hydrolysis product with the cosmetic in the market. So this is the problem. It's like the plastic. It's very difficult to establish the degradation. It's the viscosity is too large. And then we are trying to hydrolyze. Currently, we are able to hydrolyze the hyaluronic acid in water for a few hours in the perfect size. The only question is, then they need to verify the anti-aging properties with different size and so on. So this is what they are doing. But we are not thinking about adding enzyme. It's a major topic. Yeah. Thank you.
Thanks a lot.
Yeah.